fatigue setting in among the participants.
Yeah, I mean, it has been pretty intense.
Yeah, I can imagine.
So I think that as soon as, okay, I think that the live streaming has started. Great. So welcome everybody for this day of lectures. So the first speaker of today is Vishweshwar Guttal, who is giving the second lecture on bistability in and stochasticity in ecological dynamics. So please, Vishu, share the screen when you are ready.
Okay, hold on for a second. I'll do it again.
I don't see the screen shared yet. Okay.
Do you see my? Okay, no, hold on. I think I made a mistake. 1 second. Is it fine now? Can you see my slides?
I see the slides.
Excellent.
Yes. Okay.
Thank you. So welcome back, everyone. So yesterday, Simon gave a very broad overview that incidentally included ideas of bistability, tipping points, and also early warning signals. So I'm sort of going to continue on those themes today, as I had mentioned in the first talk. So I have this is the plan for the 3 talks I have. So I have sort of talked about basic bistable dynamics, resilience and tipping points, and how can we anticipate these using the ideas of early warning signals. And then today the main focus will be on spatial models. Everything I spoke on the last talk assume actually there was no reference to space. All the mathematical techniques I used was basically simple ordinary differential equations with some noise superimposed on it in an ad hoc way. So, but today, let us look at what happens if there is space. How do we understand bistable systems? Basically, the sort of one question that one can which I try to focus on today would be, can spatial patterns in ecosystems be used to infer resilience of ecosystems? That's the main question I'm going to ask. The last one, which I'm hoping will be as planned, will be to look at Okay, so today I'll just do a brief recap. I'm going to go over this somewhat quickly before I move into tipping points, right? So I give this example of large-scale continental shift. That is a classic example. This is not the only one. There are many other examples of ecosystems for gradual changes in the driver. Okay, and of course this has been observed in many systems. And to my own list of Simon added a whole bunch of other systems yesterday in his talk. So how do we? So here was the simple ecosystem model I demonstrated. The first term in this ecosystem model, where V represents the sort of total biomass density in the system, actually comprises the classic ecological model, which is logistic growth, to which we add a grazing term, which is sort of, which could be intrinsic to the system, it could also be extrinsic, such as livestock grazing. Okay, so this is a nonlinear term, and interestingly, once you add this, and for a whole range of parameter values, you get a bistability. There's a region of this grazing rate for which 2 alternative stable states exist, and which state current and there could be these switches, shifts, critical transitions, tipping points, abrupt changes at these 2 points, which are critical points or the bifurcation points of the of this simple model. So the question, the nice thing about this simple model was that we could think of the dynamics of ecosystems with bistability as a ball rolling in this landscape. So for example, the minima in these landscapes uh, correspond to the stable branches in the bifurcation diagram. So depending on the initial condition, the system could be either in this state or this state. And this this helps us understand concepts of basin of attraction and resilience. And this concept of potentials was not just for heuristics; it was also useful for uh, you know arriving at quantitative metrics that helps us make some forecasts about tipping points. For example, if the system is far away from the critical point, as in this the leftmost point here, these potentials are symmetric, whereas when you're very close to the tipping points, potentials are shallow with a low curvature and asymmetric. And this led to the phenomenon of critical slowing down and asymmetric fluctuations. Critical slowing down means that systems take now longer to return back to the equilibrium. They exhibit more fluctuations, and they also exhibit asymmetric fluctuations. Put these things together, we could come up with some metrics from the time series, and that could actually forecast. In this example, we found that if the system were to undergo a transition at this point, if we had this time series before the 1000 time units, if I could calculate simple quantities like autocorrelation at lag 1, this measures the critical slowing down. It's increasing with time as one is approaching a critical point or the abrupt transition. Standard deviation, a measure of fluctuation, is increasing. The skewness, the strength of skewness, again, which is a measure of asymmetry in fluctuation, is also increasing. Okay, now these metrics, so if you had no, if you were somewhere around 800 or the 900th time unit, so these combined metrics give us a hint that the system might be going towards a tipping point. So that's the sort of summary of where we were. I also spoke to you about how there have been many empirical studies, experimental studies, field studies. Sometimes they work, sometimes they don't work. As I also showed you, when there is huge amount of stochasticity, some of these classic signatures can be masked. Okay, so now of course ecosystems are really spatial, right? They also exhibit really fascinating patterns. These are some of the classic examples of the fairy rings found in African deserts. And these are again, patches of labyrinthine patterns of vegetation. And then, one can go on. There are more types of vegetation. Many of these vegetation patterns are often found in resource constrained systems, or systems which actually have a relatively higher amount of ecological stress. So therefore, this sort of prompted many people to think, can these spatial patterns provide some signatures of abrupt regime shifts that might happen in the future? So this is an example of clustering in the mussels, uh, and this is an example of clustering of vegetation on the seabed. So there are many, many examples, plenty of examples. So now to understand these kind of tape systems, there are three classic approaches. One is called, so the first two of them are actually both reaction diffusion systems. However, the distinction between the first and second comes from the fact that in the first system, there are no regular patterns. There are no patterning in the ecosystem. System sort of roughly appears homogeneous. There are second class of systems are regular patterning systems, and you may have heard of Turing patterns. Um, okay, and the third class of systems are there are patterns, there is self-organization, but there is no periodicity or regularity to these patterns. So there are these three types of spatial ways to think about ecosystem bistability. So I'm going to go one by one and see how we can do theory of these systems. How can we come up with these early warning indicators, followed by testing them in systems as well. reaction diffusion system. I'm going to think of spatial systems, reaction diffusion systems with no patterns. So what do I mean by that? Okay, by the way, before I go further, I want to emphasize that just 1 second, my internet seems to be fluctuating a bit. I'll see what happened.
We show also the connection is a little bit disturbed. So
1 second, I'll just see. Are you able to hear me clearly?
yeah, sometimes sometimes not. So perhaps it's better to remove the video.
Okay. I will stop
You
my
can
video.
try that.
That's a good idea. Yeah, I'll stop. 1 second. I have to unshare this for a second. Sorry. Okay, now my connection. Yeah, there's some. Yeah, it does say it's a bit slow. Okay, I'll stop my video. I'll share the slide again.
Okay.
Okay. So some of many of the points I'm going to make today, they are actually nicely summarized in this paper that many of us who work in this area came together and wrote a reviews article. So this is a reference you can refer to get a summary of the theoretical principles behind spatial patterns and how we can use spatial patterns to infer resilience. Okay, so how do we go about this? Okay, let's go back to the same model I showed you. I showed you this model where there was this simple logistic growth term followed by a nonlinear grazing term. So I'm going to assume the same term as a local reaction term, meaning in space, locally, exactly same processes are happening. There's a local carrying capacity and a local grazing rate. Of course, the grazing rate can have some stochasticity in it, which is represented by this eta c term as a Gaussian white noise. And then we add a spatial coupling term via the idea that typically seed dispersal, which is a mechanism by which one of the key mechanisms by which spatial interactions happen, is modeled by a simple diffusion term. So what diffusion term does basically intuitively is it spreads things out in space. So this is the model I'm going to use. As you do know, the mean field part of this, if there was no diffusion, if I did not have the diffusion, this system will show the same bifurcation diagram by stability. And for even when there is diffusion under a bunch of conditions, this continues to be hold. This continues to hold true. But of course, there are many subtle aspects once you add space. I'm assuming those are not in play here. So this is what we showed using a combination of numerical simulations and also analytical calculations. So I'm not going to show analytical calculations. These are actually stochastic nonlinear PDEs, right? There are methods borrowed from the statistical physics literature that we can use to compute the same things I'm going to show you today, but I'm going to focus only on the numerical simulations. So what we have done here is, over a period of time, we run these simulations and then we sort of, stress this ecosystem more and more over time by increasing this grazing parameter C. So by increasing this grazing parameter C slowly over time, we find that a vegetated system becomes a completely bare area. Okay, so and that happens over a period of, let's say, 52 years on the time scale that we chose in this model. So let's look at the top graph here. What this shows is the average biomass density. If you were to do a spatial mean of all the vegetation density, you find that it sort of gradually decays, and then by year 48 or so, it suddenly undergoes sort of a rapid shift downwards and settle down to a very low biomass density region. So we plot spatial variance and spatial skewness for these spatial images. So what we basically do is for imagine every instant of time, we are computing spatial variance, which is basically variance of all the data available in a spatial image. Likewise, spatial skewness is the skewness of all the data available for a given image. So earlier it was computed over time. Now we are computing over space. So if you plot this, what you find interestingly is that even before this nonlinear shift in the mean value happens, there is a dramatic increase, sort of, close to tenfold increase in the value of spatial variance. Likewise, the skewness actually increases from a value of close to 0 all the way to 1 and even begins to turn downwards even before the mean value has shown any significant trends. Uh, One can do these calculations again and show that even spatial correlation, the third plot here below, also shows similar patterns. So basically, the message here is that increase in spatial variance and spatial skewness, increase in spatial autocorrelations. And also, what I have not shown you here is that even in the spectral properties, which sort of, relate to both correlation and the variance, they also increase. All of these quantities increase much more rapidly than the spatial mean value alone would increase. So therefore, we can use these as indicators, early warning signals, uh, in spatial ecological systems which do not have spatial patterns. Okay, so that provides a theoretical background of how the base same ideas of time series variance, skewness, autocorrelations can also be sort of nicely transformed into can be applied to spatial systems. Now that's a theory. How do you test this in an empirical world? What do we need, especially if you have an empirical system that you really want to apply to a large scale ecosystem? How do you do this? So we need three things to be able to do that, right? One is that I need an ecosystem that has undergone a transition. Okay, and then secondly, I need to have spatial data over time at sufficient spatial and temporal resolution for this system. Finally, it turns out that there are very few, or I'm not aware of any such data available. At least, five years ago when I was looking, there was no such clean data available. So what do we do? Okay, so here is an idea. How do we test this theory? Even when you don't have, such an ecosystem that has undergone regime shift or abrupt transition over a period of time. We don't have such a data. What best can we do? I told you the theory, right? We have this state variable as a function of time. We need snapshots over time. And then we measure these metrics, for example. Imagine that instead of snapshots over time, I had snapshots over space. So the x-axis is not time anymore. It's now either space or the driver values. Okay. So this is actually an idea called space for time substitution. And it is widely used in the ecology sort of field studies. For example, if you want to understand how shifting temperatures change species compositions, how do species ranges change, one idea that a lot of field ecologists use is, of course, you can't wait for 100 years for the climate change to happen. What you do is you look along temperature gradients, altitudinal gradients, where you can find temperature gradients, and then use that space for time substitution to sort of infer or forecast what might happen in the future if the temperature were to change. So we use this same idea. If we have an ecosystem that goes from one state to other state as a function of space, can I compute these indicators and do they show expected patterns? So indeed, it turns out that there are such ecosystems. So this is an example of the very famous Serengeti Mara ecosystem in Kenya and Tanzania. And what you find in this ecosystem is the central area is sort of predominantly a grassland. However, the peripheral areas on the is actually a woodland ecosystem. Okay, so we asked the following question: if you now go from the central grassland areas towards the peripheral woodland areas, if you could think of this as space for time substitution, do you find signatures of these early warning signals as measured by spatial variance, skewness, autocorrelation, and so on, as you go from one end to the other end of the ecosystem? Okay, So we chose this very high resolution data at 30 meters, and then we classified each pixel of 30 meters as either wood or grass. And then remind you, the scale here is some, we are really talking about a few hundred kilometers by few hundred kilometers data. This is really a huge data set. Okay, so what we do is find the relationship between vegetation and the rainfall in this entire landscape. We find that, if in this landscape the grass cover sort of shows a nonlinear relationship with the mean annual rainfall, which is sort of expected from many savanna forest ecosystems. Carla has spoken about this, Simon has mentioned this. So the data here again supports the same hypothesis. So we find that the grass cover shows this highly nonlinear transition as a function of mean annual rainfall. So what we now do is, let me remind you the theoretical expectation. If the mean value of the state variable shows a nonlinear transition, then a spatial variance will increase even before the nonlinearity in the mean begins. Skewness will again increase even before the mean will decrease. Likewise, correlation and the spectral properties. Okay, the black data here, black points here represent the null model, just to show that we are not getting any statistical artifacts. So in fact, exactly as predicted, we find similar patterns in our data. So even before the nonlinearity in the variance in the mean has begun, variance has increased quite dramatically, skewness, spatial autocorrelation, and low frequency spectra. And likewise, we find this across many other transects I showed you there. So sort of this provides an evidence that one can use these ideas derived from nonlinear dynamical systems and models and apply it to messy real world ecosystem. Okay, so that sort of gives one example of how we can use spatial models of ecosystem by stability in reaction diffusion systems with no patterns. So let me now move on to regular patterning systems. Regular patterning systems are also called Turing pattern systems, and here are some really beautiful examples. I'm not going to cover a whole lot of this. Again, I showed you some picture earlier. There are many parts of the natural world where you find very nice regular periodic patterns, and these are often modeled by a mechanism called Turing mechanism, where there is a very short range positive feedback, and then there is an immediate long range negative feedback. So this combination of local positive, short range positive feedback and a longer range negative feedback causes these patterns to persist. And this paper in Science argued that these patterns may actually provide some indication of a catastrophic shift. So look at this picture here. Look at this sort of uh, diagram. So what this shows is there is a region of bistability here. Again, there's a region of bistability. As you move along this region of bistability, the spatial patterns are changing. So Reed, Kirk, and co-authors argued that these changes in these spatial patterns may offer some hints about the resilience of ecosystems. However, there are other studies that sort of contest this to show that there are so many types of these patterns, and the nonlinearity and stochasticity can sort of influence and uh, confound our interpretations. Nevertheless, but this is a very interesting idea that spatial pattern can actually offer some signatures of resilience. So now the third type of spatial patterns are slightly different. Look at these pictures. So if you look at these pictures, these two have some regularity in the spacing between vegetation. Likewise, this. However, if you look at this, this cluster is much bigger than this cluster, and then this cluster. So what you basically see here is that there is a whole range of vegetation sizes, cluster sizes that are possible. And this basically led to a whole class of new models and new studies that found some very interesting results. For example, in this paper, Scanlon et al., including Simon Levin, they found that if you quantify these patch sizes, vegetation patch sizes, and if you look at the probability density functions, they show a power law. So power law basically is a very interesting pattern, because power law often indicates that there is no well-defined mean and variance, which in other words, what this means that you can probably find really, really large values of clusters, which are impossible in simple Turing-like models. And of course, it's not only in vegetation that people have found such patterns. There are also examples of uh, such power law clusters in mussels, sawgrass, seagrass, and scrubs. Okay, of course, there is a, let me add a cautionary tale here by saying that there are many studies that also very strongly argue that in a lot of these studies, um, the statistical evidence for existence of power law is really weak. Is something to keep in mind, a very important point. Okay, and early, there has been quite a bit of research on understanding where do these power laws come from, because when you have a power law nature of any quantity, um, and when the power law exponent belongs to certain ranges, especially if that exponent is between 1 and 2, there is the mean value is theoretically infinitely large. Okay, and so is the variance. What does that really mean? How do you explain such sort of seemingly unphysical patterns? So in physics, there has been a lot of work trying to understand power laws. And in physics, the basic idea in physics is that whenever there is a phase transition, look at the figure C here, whenever there is a phase transition from one state to other state, at the point of phase transition, what's also called critical point, you see certain universal features. As Simon was pointing out, this is irrespective of the complexity of the system, otherwise, at the phase transition, the number of degrees of freedom are often very low, and so a lot of very different types of systems happen to show very similar properties, and one such property is the existence of power laws. So there has been a lot of speculation in complex systems whether the power laws that we observe in complex systems such as vegetation systems, ecological systems, and so on, are they indicative of criticality? And this is a whole field in itself, and I'm not going to really touch upon it. I encourage you to sort of read critically on this topic, very interesting topic. What I'm going to now focus more on the mechanisms. How is it that some vegetation ecosystems show irregular patterns, meaning the cluster sizes are not a well-defined average value? On the other hand, there are some other ecosystems where the cluster sizes show very clear periodic patterns, such as Turing patterns. So what's the mechanism that makes them so different? So it turns out that Hello, can you still hear me?
Yes.
Because my network said the connection lost. I'm sorry.
Yes, for a second.
Okay. So the question I was trying to address here was, how is that ecosystems of similar type show rather different patterns, spatial patterns? So one way to understand that is to look at the net interaction strength. So here a positive value means a positive feedback. Positive feedback means that the presence of, for example, a presence of a tree will facilitate the presence of another tree nearby. So there is a local positive feedback followed by no net feedback after a distance. So if you have only a local positive feedback, what you often find are these irregular shape patterns. On the other hand, if we had a local positive feedback, immediately followed by a short range negative feedback, okay, this difference leads to highly regular patterns, which are also called Turing patterns. So therefore, as Simon was pointing out yesterday, when we try to understand spatial patterns, mechanisms become really crucially important. So what are the biological processes? And often these are integrated with many geophysical processes such as water that flows in the landscape. So therefore, ecohydrology plays a very important role in shaping these feedback as well. So moving on from these, irregular. So let's go back to the original questions that I set out, which is, can we look at these spatial patterns and then make some inferences about stability? So in this paper by Sonia Kafi and co-authors, they made this following claim. They said that if you observe a power law scale-free clustering, such an ecosystem is a stable ecosystem. On the other hand, if you observed as I was trying to see my. On the other hand, if you observe that the cluster sizes are not power law, but if they are exponential, then they are more likely to be less resilient. So basically, the argument was that you can use the cluster size properties to infer something about the resilience of ecosystems. So to make that point again, what you are seeing here is a example of low stress system. This is to high stressed ecosystem. And what you're seeing here is this power law, which is bending away. Bending away meaning, this is not a power law anymore. So these parts are likely to represent more stable ecosystem whenever you see a power law or a heavy tail distribution. If you find a exponential like distribution, this could be a case of a highly resilient ecosystem. So less resilient ecosystem. So so sort of to summarize this idea of how one can use spatial patterns as signatures of tipping points. So I want to emphasize there are three types. First is that of no spatial pattern, right? When there are no spatial patterns, I also showed you empirical evidence. And then when you have these periodic patterns, okay, and then you have scale-free patterns. So when you have this no patterning systems, you can use what we call generic early warning signals, such as variance, skewness, and correlations. Those provide early warning signal. When you have periodic pattern, the very presence of the nature of the periodicity and this, usually these spotted patterns, they are often thought to be maybe an indicator that we are very close to critical transitions. On the other hand, when you have systems with irregular patterns, as you move from scale-free pattern to not so fat-tailed, thin-tailed distributions, okay, then you infer that those systems are less resilient. That's a summary of various theoretical models. So far, in my understanding, there is a reasonably good evidence that these sorts of indicators work well in real ecosystems. There is quite a bit of debate about these two, and there have not been too many empirical studies that look at these two. So now let me sort of provide a twist or a spin here, basically to sort of show that these results are a lot more tricky and complicated. So for example, in this paper that was led by my PhD student Sumitra Shankaran, what she showed was that this sort of claim that cluster size distributions or the patch size distributions that can provide early warning signal actually have nothing to do with this phenomenon of critical slowing down, which I have already sort of summarized. And in fact, the claim that loss of power law clustering is an indicator is not true. So basically in this, so loss of power law clustering is not a generic indicator of ecosystem resilience. So in fact, there is quite a bit of, depending on the model that one uses and the mechanism that one incorporates, we can actually show sort of, test the limitations of some of these metrics. So how do we go about doing this? Let me sort of try to describe how we build these simple models. So these are very different type of models. These are not reaction diffusion systems. So we call them, they're popularly known as cellular automata models. So in these cellular automata models, you have, so just one second. So you sort of assume that the space is divided into a whole large grid, like in the central figure here. Okay, and different cells either can be occupied by for example, a tree, or it could even be a mussel bed. So tree is only a representative organism here, and it could also be empty. So it could basically be one. We represent occupied cells as 1 and others as 0. And then you sort of implement certain stochastic update rules. For example, let's say if we choose this pair of a tree and an unoccupied area next to each other. So two possibilities happen. So one is that of death. So there is a probability of death here, and then there's a probability of birth here. For example, you can see that in this case, if this arrow is chosen in the simulation, this original tree is now dead. On the other hand, if the birth was chosen, there is now an addition of tree here. So there could be a birth event or a death event. Likewise, there could be additional complex interactions. So these are called facilitation or positive feedback interaction. For example, if we choose the update for a pair of trees which are next to each other, two things can happen. One of them can die, like it has happened here. So of the two here, one of them has died. On the other hand, the two together will facilitate birth of another tree nearby. Okay, and they happen with another set of probabilities. Okay, so now we can convert all these sort of intuitive rules of how birth and death happens in probabilistic terms, and depending on the neighborhood. Okay, so and in these type of models, typically there are many parameters. For example, in many of the papers I was showing you earlier, often there are 5 to 10 parameter values, and those large number of parameter values make it very hard to understand what parameter is causing what effect. So in contrast, in this model, what we have done is we have chosen a very simple model inspired by statistical physics that has only two parameter values. One is P. P represents basically local birth rate, as I demonstrated here, or even local death rate. P controls the baseline, the production and death rate. Q controls the strength of facilitation, how a plant will influence the birth and death of plants nearby. So there are two parameters, and therefore, because there are just two parameters in this simple model, one can sort of do an extensive set of simulations and find out what happens. So what we then have done with these type of models is do simulate them for a very large amount of time. So for example, I'm just showing you two representative simulations here. So on the left hand side, you're seeing a simulation for, I don't remember the exact parameter values now, they're probably low P and low Q. Okay, on the other hand, if I'm right, here on the right side, we have kept the value of P, which is the baseline birth rate, same as the previous one. However, we have a much higher value of Q, which is high positive feedback. So now we can very easily control two parameter values and then see the effect. So you can already see very clearly in this simple simulation that all else being equal, increasing positive feedback increases clustering in these ecosystems. Okay, so therefore one can study how the clustering properties change as a function of positive feedback values. So here is the classic phase diagram. So what we do here in this case is consider two cases. Q is equal to 0 represents a case when there is no facilitation. So in this case, you find that as you vary this driver value P, which is the baseline birth rate, the system undergoes a phase transition from a bare steady state, which means everyone is dead on the entire spatial landscape, and there is a continuous phase transition to a vegetated state, or also called active phase in the physics literature. And the crucial point here is this phase transition is a continuous phase transition. On the other hand, look at the value with a large value of positive feedback. You find that the response of the ecosystem to reducing value of the driver value is highly nonlinear. Look at this. For a very small range of P, the steady state density drops fairly nonlinearly, sharply. And in fact, there is actually a gap. There is actually a jump for extremely tiny values of driver values. So this shows an example of how positive feedback in space affects the abrupt regime shifts. And so unlike the sort of phenomenological model I showed you in the previous talk, so this is an example of a spatially explicit model where one can incorporate some realistic features and mimic features of bistability. So again, this would be the region of, so in this model, the region of bistability would be roughly this much or so. It's not very clearly shown here. And using this model, what we do is the following. We do simulations, and then from the simulations like this, we calculate at steady state what are all the different clusters, what are the cluster sizes, and what are the statistics of cluster sizes. So I'm going to show you one result from that right now. So what we have done here is just assume that we have chosen a value of P, which is in the middle of this phase diagram, continuous phase transition, which is low positive feedback. So I have chosen a system far from the critical point 0 here. And then you find that at this point, there is a power law in the cluster sizes. Okay, so this much simpler model than the previous ones can also reproduce existence of power law in the ecosystems. What we also find is that when the positive feedback is high, you can find these power laws even at the point of critical threshold here. So this is so this is the parameter value for this graph here corresponds to the exact parameter value where the system will abruptly collapse, and you find a power law there. So what this basically means is some of the previous claims, such as cluster size distribution, can be used as how far or how resilient the ecosystem is really not robust. For example, I'm finding the same power law distribution. Of course, the exponents are different, only marginally. Exponents are different, and yet in this case, the system is really far from the threshold, right? However, in this case, system is quite close to threshold. So one can find same cluster size distributions irrespective of how far or how close you are to ecosystem. And what really matters is the positive feedback. So positive feedback is important. Mechanisms are important. So depending on the values of the, suddenly my slides went away. So depending on the value of positive feedback, we can find, my tablet is behaving a bit awkwardly, so maybe I shouldn't do that. How do I erase this? There must be some option to erase.
Yeah, I don't know if it is I guess it's not the annotation of Zoom. Otherwise, you have to go to annotate clear.
Discard. Okay, I'll come back.
Okay, great.
Yeah, that's great. Okay. So power law in the cluster sizes therefore is not a good measure. However, there are other types of power laws that do emerge at the at the point of phase transition. Unfortunately, the resolution of this figure is a bit unsatisfactory. But what you're really seeing here is a measure of correlations, spatial correlations. Basically, what this means is that how perturbations in space are correlated across the entire landscape. What we find is that typically at the critical thresholds, correlations there are very long range correlations in ecosystems, and this manifests often as a power law in the correlation function or the power spectrum. So therefore, those could be used as indicators of proximity to critical points. So in fact, we now have a manuscript that is currently being written up where we find some empirical evidence, some supporting evidence. I wouldn't say very conclusive, but something consistent with this theory. Okay, so basically, sort of, if I have to summarize this, spatial patterns are fascinating, mechanisms matter. Therefore, however, the interpretation of the resilience from the patterns alone are subtle. One cannot naively use the patterns to make interpretations. For example, if by either by looking at the nature of patchiness or the cluster size distributions, we have shown in our papers that some of the previous simulation studies were probably finding a set of parameters where it seemed to have worked, but it's not really a general result. So, with that demonstration of various simulational results, let me sort of briefly allude to what kind of analytical techniques can be used in these studies. So, unfortunately, I'm not going to go into much detail. So, for example, we can take the cellular automata model I have described. One can write down a mean field approximation, and the mean field approximation makes a very nice set of predictions about existence of critical points, whether the phase transition is continuous or discontinuous. It often underestimates the values of positive feedback one needs to find a discontinuous phase transition. One can also do something called a stochastic demographic approximation. Again, I'm not going to go into these details. I'm just going to mention to you, and I would, if any of you are interested more in this, feel free to get in touch with me. I will be happy to share some of our own labs, as well as more general manuscripts that sort of describes these methods. So basically, there are a whole variety of methods one can use. So classically, the idea of a mean field approximation is that we have a well-connected system, and I'm looking at a deterministic limit where n, the number of sites are infinitely large, the number of individuals are infinitely large. However, we know that real ecosystems have finite numbers. How can we know the effect of those finite numbers? So to do that, something called a finite size expansion, which is a very powerful technique. And there are methods called Fokker-Planck equation, Langevin equation, using which one can find the steady state distributions. Okay, so often in this approximation, you don't have a deterministic equation of this type. What you have is a stochastic differential equation of this type, and the stochastic differential equation would then need to be solved using the methods of Fokker-Planck equation and Langevin equations. And in some cases, the stochastic differential equations can also capture the effect of the system sizes that you are considering. So I'm not going to go into any further details of this. So I'm coming to sort of end of my talk. So a lot of the work I presented from my lab today was done by these two PhD students. They have finished their PhDs now. Really very fabulous work done by them. And then also many collaborators, in particular Sonia Kefi and her students Alex and Miguel. And of course, there are many more collaborators and funding that has made this work possible. In particular, Amit, Ashwin, and Stephanie, Andy Dobson, they were all involved in some of the work I presented today. So finally, before I take any question, as I had mentioned, so I'm going to talk about intrinsic noise and bistability in collective. So that's the sort of, prelude to tomorrow's talk, how we can actually use similar ideas of noise and bistability, and how noise has some very counterintuitive effects when we study collectives, not of plants and trees, but instead of animals. So I'm now happy to take questions. I think we have quite a bit of time, so I'm happy to go into details of anything that I have sort of skipped over.
So there are a few questions in the chat, which I read
Sure.
for you.
I will also try to open.
I just to remind that if anyone wants to ask a question, please use the.
Oh, quite a bit of. I should have seen this midway through. Sorry.
Okay.
So it doesn't pop up on my window even when somebody chats.
Sorry?
Okay, so go ahead with the questions, please. Yeah.
Yes. So I was just saying that if anyone wants to ask a question about, can use the raise hand tool. So I'm gonna start reading the question. So the one question from Tuan is, so there is no universality for spatially correlated systems found in ecology in general?
So is there no universality for spatially correlated? It's a tough question. I can only give a lousy answer. See, clearly, there are many common principles and common patterns, right? You look at zebra code patterns, or these some of the patterns I showed you in the beginning, right? For example, sorry, if I can. For example, sorry, not these. Yeah, look at these patterns, right? Some of these patterns are even the code patterns in zebra. We now know that the code patterns in many animals and this kind of vegetation can both be explained by mechanisms such as Turing instability, Turing patterns. So there is generality, and there is universality in the spatial pattern formation. But there are also some details that are important. Certain additional parameters like strong positive feedback and the scale and the strength of positive feedback can confound these results. Therefore, I think one has to use a combination of these general mathematical theories together with the mechanistic approach. I think one has to use a combination to sort of understand the limitations of universality here. I don't know if I answered your question well.
Well, there is no sign from. Yes, thanks you for the answer. So there is a question from Miguel Rodriguez, please.
Yeah, I'm thinking a little bit of the lack of data sets that you mentioned to test these principles, and I was thinking if maybe land covers superposed, like land cover maps superposed with carbon sequestration maps could be used, or that's just too coarse a resolution. Because for both of them, there are contemporary distribution and also historical distributions around the globe.
So I think so I spoke about three types of spatial patterns, right? One is the first one being where you coarse grain so much that you don't know the spatial patterns anymore, right? The second one is there are very fine scale spatial patterns, So those images I showed you, those were patches over tens of meters of approximately those scales. So the same with third one as well. The irregular patterns, they also need to be observed on those scales. I think the first one can be done using some of the data sets you mentioned. So those Landsat kind of data sets,
right?
Which we are currently working on as well. We are using a whole bunch of satellite-based vegetation metrics to not only look at the patterns of various spatial metrics, but also can we construct model from data? Can we derive model from data as well? So we are also looking at those questions. But if you want to look at these fine scale spatial Turing-like patterns, I could be totally wrong, but I think that the data sets that clearly show evidence for the theory are lacking right now.
Thank you.
Great, we have time for more questions.
I think there are more questions on the chat box. Actually, I can see them.
Yeah. There were a few questions about the null model.
Yeah.
But what
is the null model? One without bistability. Yes, that's right. So the null model. So I don't know at what instant you asked me this question. So I think you were maybe referring to the null model I had in one of those slides. So for example, what one can do is imagine that the density, let's say there is a huge spatial landscape, and you find that 40% of the area is covered by trees, right? So 60% is bare ground now, right? So now how do I know it's driven by positive feedback? That's one case. So what one can do is you can create a null model which maintains the same cover in the landscape, 40%, but just randomize the distribution of the entire trees. So the clusters that will be formed in the randomized data set will be entirely not because of any interactions in ecology. They are entirely because of the density itself. So that's one way to arrive at null models for some of these kind of questions. Yes, that's right. So it will not have bistability. So it's just entirely, I made up the data, right? So,
good.
And so the second, there's a question on what causes the sharp negative feedback. I think it's this Turing system, right? So you find that there's a positive feedback for short range, which becomes negative feedback for longer range. So what happens in these systems is that, so for example, if these are clusters of trees, so there is they draw water from neighboring regions. So the neighborhood of trees will have higher water retention. What that also means is that the slightly farther away from this cluster will be devoid of water. So the water is sort of conserved. If you were to think of water as conserved to some on very short time scales, so the rainfall falls homogeneously in a landscape. So there is a cluster of trees. They not only retain locally well, they also draw water from the local neighborhood. So because of which at slightly larger distances, there will be a lesser amount of water than the average. So that causes the negative feedback at this slightly longer range distances. Oh, somebody has answered the question, so that's great.
So yes, it's the beauty of the chat. So there is another question by Zore, who is asking what does feedback bifurcation mean in ecological modeling?
I don't think I understood the question. What I said was the positive feedback can cause new types of bifurcations. So when we have weak positive feedback, you typically have continuous phase transitions or continuous transition from one state to other states. On the other hand, when you have strong positive feedback, the transition can become abrupt. So that was what I was saying. I was not sure if I used the word feedback bifurcation though.
Great. So we have, if there are no more questions, I don't see any hand raised. So great. So there will be the last lecture by Visho on Thursday. So 2 days from now.
Yeah.
I think you will have opportunity to ask further question. Now we are going to move to let me thank Visho again for the nice lecture.
Thank you, Jacob.
Now we're going to move to the breakout rooms, and we're going to be back in 15 minutes for a seminar by Flavio Marchitti. So,
See you everyone. Bye.
Bye. Thanks. Okay, welcome back. I think everybody is here because I don't see the breakout rooms open, so I guess they are have already been closed. So before we start with the next slot, let me remind you a few things about the program. So after after the next talk, there is going to be a long 1 hour break, and then we're going to have other 3 lectures. Then tomorrow there will be the colloquium by Professor Ned Wingren, followed by a roundtable, and on Thursday we're going to have other lectures and roundtables. So just to clarify something about the roundtables, so the roundtables are going to be a discussion among 5, 6 panelists who are like people from the pool of the lecturers who taught at this school, and there will be a discussion among them, and there will be the possibility to ask a question and to participate, of course, to the discussion. So that's what the roundtable is going to be about. And as you see from the program, we're going to have 3, and well, there is nothing you have to prepare for them, just popcorns, and then watch the discussion and participate to it. So without further delay, it's my pleasure to introduce Flavia Marchitti, who is currently a postdoc at the University of Campinas in Brazil. And today she's going to talk about the phylogenetic patterns and how to understand them using microevolutionary models. So thank you very much, Flavia, for being with us and for giving this seminar.
Thank you, Jacopo. Thank you for the invitation. And please check if the share screen is working. Is it fine? Okay. So I'll be talking about just let me put you on the side here, sorry. So I'll be talking about these phylogenetic patterns using an evolutionary model. And I've been working with this topic in the past 5 years, but I'm very interested in many topics explained and exposed here in this winter school. And I'll be clear about that in the next slides. So I'm very grateful to thank lots of collaborators and mentors that I had during my lifetime. The list is longer than you can see here, fortunately. And I'm also very happy to have the finance of supporting grants in Brazil and to have the opportunity to go overseas to study. And this is not a great time for science in Brazil, and maybe not for many places around the world. So I'm very grateful to be financed by FAPESP and CAPS here. And so during this winter school, we've listened a lot about coexistence between different species. How can we have different species sharing resources and still coexisting in the same area or in the same environment? And I'm very interested in this topic as well, but I'm also very interested in how these species form, how they appear the first time. And studying the species formation and how these species coexist, I've been working with geographic modes of speciation, and I've been working with neutral models to form a backbone to understand non-neutral models, how we can learn from neutral models from the absence of competition, absence of any other forces, to understand how these things will change the species formation. And the species diversity form patterns, for instance, patterns that can be observed in phylogenetic trees. we see different species forming, and they are related to each other in such a way that can be described by these phylogenetic trees. And there are many forces shaping and process shaping these patterns that we observe. For instance, how species that coexist in the same area, how they diversify in different species. And what are the signatures that we can have, for instance, from species that don't share the same area. And with this, the main line of my research nowadays is to understand how this we can link the macroevolutionary patterns and the microevolutionary forces. And this fills a gap that exists in the literature called the micro-macro gap. It's too hard for me to say that, but I think you got it. And when we talk about speciation, when we talk about diversification, usually we think about geographic configuration. We usually learn that the species form when you have some barrier that separates individuals in two different areas. And these individuals accumulate differences, and once they are together again, they don't recognize each other as individuals of the same species. This is known as allopathic speciation, and usually is invoked to explain how a species form. But there's another form, and this is likely the process that happened with the chimpanzees and bonobo apes in Africa that were divided by the Congo River and speciated. But there's another form of speciation called sympatric speciation. So the sympatric speciation happens, or doesn't happen, it depends if it exists, when individuals share the same area. And there's a debate in the literature if this is an important force for speciation to happen, if it really exists. But I think before this discussion, before this debate, we need to learn how it happens, if it happens, how it happens, and what are the signature it leaves for us to understand that it happened. So that's why I've been working with models in sympatric speciation to understand how we can then deny this is the process that is shaping evolution in many species or in many phylogenetic trees. And one of the most well-known sympatric speciation models was proposed by Dickman and Bell in 1999, where they used a model where they included assortative mating, which is the individuals, they choose sexual partners based on how similar they are. They also included gene linkage between the mating character and the ecological character, and also included competition, the model, to understand if individuals sharing the same space could then split in different species. And they showed that with these ingredients, they could observe the species formation, these bifurcations on the ecological character. However, earlier than that, in 1991, Higgs and Derrida, also known as the Derrida-Higgs model, proposed that sympatric speciation could happen under a neutral model. And maybe some of you will think, oh, neutral models are useless. They don't show us anything. But I think we can learn a lot from neutral models to then understand non-neutral models. And that's why I like this model a lot. So the Derrida-Higgs model proposes that we can define individuals by a very large genome, as large as infinite genome, that have each gene in this genome can have a +1 or -1 allele in their positions. And they show, and I tell you how, a population of M individuals can then split in groups of different individuals that we will call species here. And they do this require a simple thing, that is a minimal similarity between individuals to reproduce. They do not prefer to reproduce with the most similar, but they must have a minimal similarity to reproduce. So what happens in this model is that we have two individuals that are haploid individuals. So they have only one string that defines the genome. The genome is biallelic, as I told you. And we can compute the similarity between two individuals for the reproduction. So here, individuals alpha and beta, they have similarities in some positions, here and here, but they have some dissimilarity in other positions, for instance, in here. But if they have a minimum required similarity for the reproduction, they can then mate and form an offspring for the next generation. And this offspring will have the alleles from their parents with a probability of a half it comes from parent alpha and a probability of a half it comes from the parent beta. And 8 of these alleles will have a chance of mutation given by mu. So here, for instance, the individual should be a positive one in this allele, but it should be a negative one in this allele, but it became a positive one because of the mutation. And with this, I should stop the video first, sorry. With this, starting in a very, sorry, I don't know how to reproduce it again, but starting with a very high similarity, all individuals are very similar. Passing the time with the sexual reproduction and with the mutation rate, we get individuals that have a lower similarity between each other than a given point that is defined by the minimal similarity that we define for the reproduction. So these individuals that have dissimilarity between each other is that where we see a species formation. So we get individuals that cannot reproduce to each other. And so with this simpler model, the Hida and Higgs have shown that with the sexual reproduction, with a minimal similarity between the sexual partners, and with a very large genome, as large as infinite, we can observe that we'll have groups of individuals that are dissimilar to each other and cannot reproduce to each other. We can observe speciation with no geographic structuring here, so in a sympatric mode of speciation. And as you can see, the infinite genome is a very large cost paid here to have a speciation under a sympatric situation. So something that started with Marcus Aguiar in the past 10 years, he decided studying the Hida-Higgs model in a modified version. So we try to work with a model where it's more real, more close to real situation, where genomes are not more infinite, they are like finite genomes. So we have a genome that have like B lossy, and exactly as in the same model of the Hida-Higgs, we have two individuals that will reproduce. They must have a minimal similarity for the reproduction, and there is a mutation rate that can change the offspring inherited allele. And with this, Marcus shows that it's possible to have not infinite genomes and still have sympatric speciation. So he shows that as we have lower genome sizes for a fixed number of individuals, we must have a lower mutation rate as well to have speciation happening. And the other thing that is important is that for a given fixed mutation rate, as the genome size increases, the lower the number of individuals required to speciation to happen under a sympatric mode. And this is something important. We show it's not required anymore to have an infinite genome for the sympatric speciation to happen. So it's more close to real world. And also, something that Marcus decided studying was to understand how we can enhance, how can we force a speciation, putting some structure on the spatial area where individuals live. What about if we decrease the sympatry level somehow? And he has shown that if we put a radius around the neighborhood and in the neighborhood of a focal individual for the reproduction, forcing this individual to look for a partner, for a sexual partner around this radius, and we can change this radius for a small radius, can we observe still speciation happening? So here we are decreasing the sympatry level, but we still have individuals in the same area. And he shows that when we have a mating radius, a smaller mating radius for looking for the sexual partner, we observe that the genome size required for observing speciation is lower. So we can model the sympatry somehow using small genomes and some structuring on the space for observing speciation. And more than that, he shows that in a given amount of time, if the radius is small, we observe more species, and these species are more structured in the space than if we have a larger radius in the same time observing this one. So this was very interesting and very, very important to help us to develop the other research that we did in the past years. And one of them was trying to do that thing that I showed you before, to look for some relationship between species and how this relationship between species could be related to the geographic mode of speciation. So we observed that species were forming through time in a spatial model. And we also observed that, that I told you before, when we have a spatial configuration, we can observe that as larger is the radius of the individuals to look for partners, the less species are formed. And also, when we observe a smaller genome, it takes a longer time for species to form and find an equilibration of the number of species formed. And this was very important, and we were interested in relating these observations with the phylogenetic trees of the species that were formed in these spatial areas. And so we developed methods to construct the phylogenetic trees under this speciation model and this individual-based model. So we developed a method where we could record all the speciation and extinction events during the evolutionary time. So we could say which species came from whom, and if, for instance, extinction events were happening, and how long it takes to one species to form and then to form another one. What are the times of these lengths, of these branch lengths? And developing this method, we can also annotate more information during the evolutionary model and have a more concise information by the most recent common ancestor. And we observed that these two methods, which are the true phylogenetic tree represented here by recording all the speciation events and annotating more concise information, the most recent common ancestor, we could use information from the phylogenetic trees to compare these two methods. So using, for instance, metrics to describe the branch lengths. For instance, we have the an acceleration metric, or the gamma statistics that define us, that the phylogenetic tree for us, that describes to us that the phylogenetic trees has longer branches or smaller branches, is more accelerated or decelerated phylogenetic trees, and also information about the imbalance of these phylogenetic trees. How low is the imbalance of these phylogenetic trees, and how caterpillar is the format of these phylogenetic trees. So using these metrics to describe the phylogenetic trees, we could see that annotating all the events, or also annotating more concise information by the most recent common ancestor, was very similar. We could use the most concise one. And here I show you very happily that this annotating all the events, so having the true phylogenetic trees, presents to us the same, using the most recent common ancestor, presents us the same traits about the gamma statistics, that is about the branch length, and also about the balance of these phylogenetic trees. So these methods are very similar, and it helped us a lot to get information and do simulations more fast. So as I told you, I was interested in understand how these radius of the mating range could change the phylogenetic trees. So I was interested in understanding how parapatric and sympatric speciation were different in terms of the phylogenetic trees formed after the process. So just reminding you, we observed that we would have less species under a larger mating radius, and also would take a longer time for small genomes to form the number of species that we observe in the calibration time. And something very interesting happened when we did the simulations after that. We observed that there is a signal here observed mainly in the radius, meaning the parapatric sympatric level of the spatial use, where we observe that as more sympatric is the phylogenetic tree, the larger the radius, it means that more TP are the phylogenetic trees. They have this format here. The smaller the radius, it means the more parapatric is the speciation in our model, the more steamy are the phylogenetic trees. And we also observe that the genome size, we can observe here the same radius and different genome size that increases from here to here, from orange to blue. We observe that it affects both the acceleration and the balance of phylogenetic trees. And we compare this to empirical phylogenetic trees, adaptive and non-adaptive phylogenetic trees. And we observe that we could observe some patterns in our data. And observing especially, these are the trees that we worked with, and we observed that the higher the genetic flow during the speciation time, the more balanced were the phylogenetic trees. While the less genetic flow happened during the speciation time of these empirical trees, the more unbalanced were the phylogenetic trees. And this was very related to the radius of the mating range that we had here. So it seems, and in our model, we can observe some signatures of the geographic mode of speciation. And maybe there are some signatures out there in the phylogenetic trees that we have in databases that we can relate to the geographic mode of speciation. So I was very interested in these results that we have about the sympatry and the parapatry. But I was also interested to understand, okay, how can we tell about the barriers, about islands, about teams? If we have some barriers, defining our space? Can we detect something in our phylogenetic trees? So if we have allopatry, so going back more close to the real world, if we go to the allopatry, can we observe some pattern in the phylogenetic trees? And by the results that I've just shown you, I was expecting that as we impose allopatry, as we decrease the radius, as we increase the structuring in the space, I was expecting that we would observe that phylogenetic trees will become more steamy. So I would expect that given allopatry speciation model, I would have more steamy phylogenetic trees. And then we performed our model in this structure space, and we observed that, contrary of our expectations, we observed more balanced phylogenetic trees, and I think here is where we can see it more clearly. So we observed that groups of species, sometimes called deans, were observable from the phylogenetic trees. And something very interesting could be detected when we put the balance of these phylogenetic trees according to the number of species. It's well known that the balance of phylogenetic trees, it varies with the number of species, even in a normalized metric. But still, when we did this, when we put the results of our simulations here, we observed that for a given number of species, those that were structured in more deans had a smaller balance or unbalanced metric than those not structured in deans, just in one dean. And it was interesting to observe that for the some Hawaiian species, such as the silver sword alliance, the tetragnathus spiders, and the Sticta lichen, that are very splitted in different islands in the Hawaii, we could relate it that these species have been formed in different deans. They did, and it could be detected by their balancing, by the balancing of their phylogenetic trees. However, for some other species, for instance, for the finches, for the diving finches, we didn't observe we couldn't relate the balance of their phylogenetic trees with the many islands where they formed. So maybe, and we explored this in the paper, maybe this information was erased from the phylogenetic trees. And contrary to our expectations, the alpha value that is related to the branch lengths of these phylogenetic trees didn't change a lot. It's true that the variation did, but we didn't see any change in the mean value of the alpha value. And so it was a surprising result. And when running these models, we observed that we are not only creating new species, we are also losing some diversity somehow. And the diversity was being lost by different methods. And the most well-known and the expected one was the extinction. So some species were formed and passed some time, they would disappear as the number of individuals decreased. And other processes such as reversal and fusion, that I will call broadly as hybridization process, we observed that some species somehow were being splitted and then joined again in another species. So we were forming species, but then we are losing them by hybridization. And together with the student, we decided to study how frequent these things were. So the species could be defined somehow as the group of individuals that have some genetic flow between them. It is not necessarily a direct flow, but it must be somehow some flow between different individuals. For instance, this one and this one, they have a genetic flow through this third individual here. And so a species could form, and the individuals were linked by genetic flow. And we observed that the hybridization could be defined that when we have a species formation, such this blue one, but then reverse it somehow to the species that it came from. So this is the most typical hybridization process that we usually call fusion here. And the extinction is really the disappearance of a group of individuals that by fluctuation, they just disappear. So we could have in our phylogenetic trees these three processes. The extinction, where individuals disappear just by fluctuation of the number of individuals in that species. The fusion, where a species splits into two and then fuses into one again. And the reversal that requires two speciation process, which is easier to see here, and then a fusion between the oldest one with the new one. It's not a fusion between the newest species, but a fusion, a hybridization between the most recent and the most the species that originated these two. So what we could see is that extinction happens all the time, and it's basically independent of the genome size. We see some difference here. We observe that smaller genomes have a lower extinction rate, but it's not so different as, for instance, fusion and reversal. So fusion and reversal, they happen, these hybridization modes, this hybridization process happen all the time, but fusion happens more and reversal gets an equilibration. And we observe that the fusion is more common as a smaller the genome. And we also observe that, as expected, the population size during the extinction happens more frequently in small population sizes. So just before the event happened, if the event was an extinction, the population size is likely a small population size. While fusion and reversal, it happened in any population size that we had. And the extinction and fusion and reversal didn't change a lot on the branch length of the species where that event happened. More frequently, extinction could happen in species that were very old or very new, and fusion and reversal seems to happen in more new species, more recent species. So I've shown here that the neutrality helps us to understand how the genetic fluctuation and population size fluctuation, also known as genetic drift or ecological drift, could lead us to structures of phylogenetic trees and how this extinction, fusion, and reversal could happen during this process. And it helped us to understand, to form the theory, understand more broadly how we could then develop non-neutral models, understand better non-neutral models. And in our group, we did this in two different ways. I will show you the results by Deborah principally. I think she's participating here. And also a work that is being developed by my student nowadays. So we had the backbone to understand how genetic interactions, for instance, the metanuclear interactions, could reflect on the phylogenetic trees. And we've been studying as well how the cooperation evolution is linked to the diversification or to the formation of new species. So I'll start with Deborah Principe work. So they developed this very nice model where the individuals, they have not only the normal genome that is called the nuclear DNA, but they also have the mitochondrial DNA. And usually the mitochondrial DNA is inherited by the mother. And these DNAs, they must have some compatibility, the nuclear and the mitochondrial one, because the respiration, a very important process, is it's a link between the proteins and everything that is necessary for the respiration process made by the nuclear and the mitochondrial DNA. So they must have some compatibility between them. And using this, the difference, the compatibility between these two DNAs, Deborah defined that individuals that have a better, a smaller distance between these genomes, or a more coupled DNAs, they would have a larger fitness that should be defined by a function. And again, individuals can be defined in space. And something very interesting that they observe is that as we increase the force of the selection, and the force of the selection is defined by the how broad is this fitness here. So as we increase the force of the selection on the genome and the nuclear and mitochondrial genomes, we observe less species. So here, the smaller the value of a sigma here, the larger the selection force. And also a species that were, and the NI value here is when we have no interaction between the nucleus and the mitochondria. And also they observed that extinction were more frequent when we had selection, but not exactly in the higher selection, in the the greater force of selection. They observed that intermediate values of selection provoked more extinctions in this model. And they also observed that the phylogenetic trees were affected by the force of selection between mitochondria and the nucleus. And so comparing with when we have no interaction between these two genomes, when we have a selection force, the balance of the phylogenetic trees was, the unbalanced metric of the phylogenetic trees was lower, and also the phylogenetic trees were more TP. And this is very interesting, and I think this is how we can summarize one of her results. And when we compare the coupled part of the DNA under a strong selection, and when we have no interaction between mitochondria and the nucleus, we observe that the mutation rate present on the genomes that were linked decreased the mutation rate of the mitochondria and of the nuclear DNA. And also comparing inside the same individual, they observed that in the end, the selection is decreasing the mutation rate, is purifying somehow the DNA formed in this species. And something very interesting about the mitochondrial DNA is that they can be used in biological environments and in real environments for identifying species. And this is something that they have shown, that the mitochondrial DNA has a higher matching with the DNA of the nuclear part. And so someone could use the barcoding, the DNA, the mitochondrial DNA for barcode, yes, but it's not necessary, and this is something very impressive. It's not necessary that these two genomes interact. Even when there is no interaction, there is a mating ratio between these two DNAs. And just to finish with my last work here, I've been working the last years to understand how cooperation evolves. And this is something impressive that happens in the most variety of species. You can observe, for instance, Hymenoptera that interact with each other. You can observe mammals that cooperate with each other. And understanding how cooperation happens in nature help us to understand how we can force cooperation in human populations. Maybe it's a dream, but it's something that we could say that we can learn for nature. So usually the cooperation evolution is studied using evolutionary game theory. So in evolutionary game theory, we say that we have individuals that cooperate and some individuals that don't cooperate. They're called defectors. And in a given amount of time, depending on the interactions between these individuals, we will observe that one of these strategies will be stable. And usually we study how we studied systems where the defection is the stable strategy. And one of the most well-known games is called, it's represented by this payoff matrix, where we have the two strategies, cooperators and defectors. And we say that a defector receives a payoff of 5 when interacting with cooperator, and it receives a payoff of 1 when interacting with another defector. And the cooperator here receives a payoff of 3 when interacting with another cooperator, and the cooperator receives 0 when interacting with a defector. And thinking that these columns here represent populations, we can see if the strategies in the rows can invade the populations defining the columns. And usually in this type of game, call it the prisoner's dilemma, what we observe is that independent of what happens, the defection is the stable strategy, because the defector can invade a population of cooperators, and a defector can invade and be stable in a population of defectors. So this is the final result usually, and something that has been studied in, I don't know, in the last 30, 40 years. I cannot calculate things now, but in many years, and is how we can have the cooperation evolve in situations where we are expecting the defection to be stable. And Novak and many collaborators have shown that the spatial structure can be an important force for establishing some cooperation where only defection was expected. And so using this simpler form of the prisoner's dilemma, where we have 1 here and a p-value that is varied here, Novak and collaborators have shown that even for values where we are expecting defection only, we could observe some cooperation if the interactions were defined in the space. If the interactions and the payoffs gained by the individuals were defined by a neighborhood. And even for higher values of p here, where only defection was expected, because of the spatial structure, we could observe some formation of this group of cooperators. And so we decided studying these in our model, in our individual-based model. So if we've included in the end of the genome, these 2 traits, the cooperation and the defection, that defines the payoff that each individual gets in their neighborhood. So again, using the same payoff matrix used by Novak and collaborators, we observed that the spatial structure could still form species under this non-neutral model, but under a game situation where individuals have some fitness because of interaction with others, the number of species formed is usually smaller. And the cooperation frequency maybe is not clear here because I've started in the number 1 and not in the number 0. But when we start with the same number of cooperators or the same frequency of cooperators under a neutral game or the absence of game or under a neutral model, we observe that the cooperation keeps the same frequency for the whole time. While for the game model that is also spatial, as Novak did, we observe that there is a value of P which defines values of P where we were expecting defection, but we still could observe some cooperation. And these values are different of those observed by Novak. And I'll explore more that. We also observed that species could form, so we could draw their phylogenetic trees. And this is something very impressive. We observed a very different balance during the species formation and also more accelerated phylogenetic trees. So we could detect somehow, as Deborah did in her work, that when we include some selection here, when we include some fitness here, there are things that are detectable in the phylogenetic trees. And so the phylogenetic trees here are reflecting how the spatial structure and cooperation evolution affect the phylogenetic tree. And something we are still interested in is understanding how the absence of the structure can affect not only the formation of species, but also the cooperation evolution. And so doing a greater neighborhood for the species to the mating range and also for computing the payoffs of these individuals, I'm interested, and Louise is developing this work, my student. We are interested in understanding how species form, and this is not anymore a neutral model, so species cannot form, but we have many clues that they will form. And we are interested also if there are patterns in these species that are formed. Do we observe more defectors in some species? We observe more cooperators in other species, and how these leave signatures in the phylogenetic trees. Is it a conservative trait? Can we relate the cooperation or defection as an ancestral trait of a given group of species here? And so this is the kind of thing we are exploring now. And in the end, another student of mine will be studying how the cooperation and the hybrid, the cooperation can affect the extinction and hybridization rates. Is it more frequent that cooperators will be extinct? Is it more frequent that hybridization happens in sympatric speciation or in parapatric speciation? So this is the kind of questions that I'm interested in now. So just to summarize, I think I have 2 minutes here. I've shown you that competition or natural selection is not necessary for sympatric speciation, and this is a result of the Hida-Higgs model. And I've shown you that even in finite genomes, when we have a given genome size, a given mutation rate and a given number of individuals, we still can have sympatric speciation. And spatial structuring, when we define a mating radius for the individuals, we can have more species. We can find signatures of the geographic mode of speciation in phylogenetic trees. And I've shown you that allopatry and parapatry affect mainly the balance of the phylogenetic trees. And almost in the end, I've shown you that extinction and hybridizations, they can be characterized in neutral models, and we can then explore non-neutral models. They can be interactions between inside the same individual, or they can be through interactions between different individuals. But all this knowledge that we accumulated studying neutral models has helped us to understand and to perform the non-neutral models. So I'm ready for any questions from you. Thank you so much.
Great, thanks a lot, Flavia, for the very nice talk. So we have time for questions. So there are a few from the chat, which I'll start reading for you. And then if anyone wants to ask anything, can raise the hand on Zoom. So Elvira is asking, do you use empirical phylogenies without dating to compare with the simulation of these models?
The empirical data without dating to compare with simulation. No, it's with dating. We use with dating.
And then there is Margaret, who is asking, I noticed you made use of the random forest model on the phylogenetic trees. I wish to understand why you made that choice. Also, did you try other models? And what were your discoveries?
Oh, maybe I don't know what is a random forest model. I don't know.
Yeah,
I don't know if maybe she can ask. Maybe.
Yeah, Margaret, please.
Yeah. Good afternoon. Thank you. Can you hear me?
Yes.
Okay. Yes. If you check, I don't know the slide number when you were showing the inheritance from the phylogenetic trees is actually a random forest model that you use to model those trees. I don't know if you can go back to the slide. I can't really get the exact slide. Yeah, those branching that you had, like.
Is it in the beginning?
Yeah, kind of in the beginning.
I'm sorry.
Yeah, before this slide, yes. Backward. Okay, yeah. Here, yes, exactly. Yeah.
And this one?
Yes, under the, yeah. And there's a place that is actually stated that we use a random forest and abrupt model to actually get the models all sorted out. So I actually wish to understand why you chose the random forest.
Oh, so this is how we represented this method that we call it. It's also known as the most recent common ancestor. And I don't know if it's exactly the same thing of the random forest model, but we get individuals of our simulation, and then we trace back from which species it came from. So for instance, this individual here, let's say the yellow one here, maybe it's easier. The yellow one here, when we trace back this group of individuals that are represented by this guy here, and when we trace back, it came from a speciation that happened just in the beginning. And the blue one here, we can see that the individuals of the blue one here came from yellow individuals. So when we join the information, so we have the information based on this individual here that the speciation happened 2 times before the present time. So we can say there is a distance between the blue and the yellow one that happened 2 times before now. And this is just like following back the species that identifies the species of an individual and which is the ancestor of the species of that individual. I don't know if it's the same of the random forest. I didn't know.
Yeah, it is, but I think in further slides too, it was actually stated specifically. I saw it, I can't really remember the slide. But all the same, I've sent you a private message and I hope you reply because I want to know your choice of model. There are actually several models that can do this, but yeah, we know generally the random forest tends to be like the most convenient and most accurate in terms of what to do. But there are also other models that will give a better choice. So I just wanted to understand the choice. But all this in that's okay. Thank you.
So just one more thing, maybe it can help you to understand what was our choice. So this information, so using all the events, so recording all the events takes a lot of time and takes a lot of information that we need to record in our computers. So doing this helps us a lot. But we also explored, for instance, genetic distances
Okay,
by
yes,
different metrics.
this slide exactly. We check the first slide. You said the normalized random forest. Yeah, that was why I had to ask why random forest. Why random forest exactly?
So yeah, because we have the information of the genomes of the individuals, we could do this genetic distance between individuals of different species. But we've noticed that when we compare to the real phylogenetic trees that is made during the evolutionary process, when we can record all the events, we observe that the phylogenetic trees made by the most recent common ancestor were more similar to the real phylogenetic trees. That's why we decided to use this method to construct our phylogenetic trees.
Okay. Thank you so much.
You're welcome. Thank you.
Great. We have time for more questions. Since nobody's asking, I can ask my question. So in one of the first plots you showed about the model where you compare with the empirical networks, you had adaptive and non-adaptive networks. I mean, I'm not sure how
Trees. I also say networks, but it's trees.
Trees, sorry, trees, sorry. Yes. I mean, there is gene flow, so they are also network in some sense. But,
yeah.
so you have the adaptive and non-adaptive. So I'm not sure how these two are classified, but I would have, I mean, the word adaptive suggests that there should be some, at least difference in the structure, while it seems that you didn't see any difference between the two. So I was wondering.
I can go back to that slide as well. But it's true. So when we decide to compare to adaptive and non-adaptive radiations, it's below here. It was because we were evaluating our phylogenetic trees. Let me show here. We were evaluating our phylogenetic trees under this when they reached the equilibration time. So we were interested in this radiation process. When it takes longer time, you see that they don't form new species, so the phylogenetic trees will reach somehow some structure that is somehow fixed. And we decided to study how radiations what are the signatures during the radiation. And then when we go to the literature, you only find or mainly find adaptive radiation. And maybe it's because we believe that the main process happened under adaptive radiation. So usually when they say adaptive radiation, they are informing us there is somehow some selection during the process. And the non-adaptive radiation, so this information of adaptive and non-adaptive radiation were also got from the literature. So as the authors inform us, oh, it's adaptive, it's non-adaptive. And if you see in our table, oh, maybe I should put in the presentation mode. We have usually more adaptive than non-adaptive networks. And it's true, we can see some structure here in the adaptive phylogenetic trees that are the triangle ones. That is given by the natural selection, not by the spatial structure. But it was interesting to understand and to see that maybe the genetic flow that can be defined as low or high defines better how the structure of the phylogenetic, more than if it's adaptive or non-adaptive. Maybe the genetic flow is the most important thing here. And we made this classification of the genetic flow based on the how many islands or how was the structure during the radiation process.
Mm-hmm.
Did I respond to your question? Sorry.
Yes, no, it's I was, I mean, I think it was surprising that there was no difference between the adaptive and non-adaptive, right? Because they both match the neutral case.
Yeah, and I think because we have this information that maybe the space, the spatial structure is very important, we have we are more prepared to evaluate the non-neutral models and understand what are the signatures on the non-neutral models that are not given by the space.
Yes, pretty clear. Great. So is there any other question? Okay, so if not, let me thank Flavia again for the very nice seminar.
Thank
and we are now going to take a break for 1 hour and a half. So it's going to be a long break. You can even take a walk out of the screen and go out. And we are going to start again at 3:45 with the second lecture by Alvaro Sanchez. So thanks to everyone. See you in hour and a half. Bye-bye.
Bye-bye.
Welcome back, everybody. So in a couple of minutes, we will start again. So I think many people typically connect at the last minute, so we'll start in 2 or 3 minutes. Great. So we are about to start. Before I introduce the next lecturer, let me remind you to check frequently the program for the next 2 days. So in particular, tomorrow we are going to have a colloquium by Ned Wingren on modeling microbial diversity, and I strongly suggest to attend that activity. Again, a reminder, if you go on the website in the program, you will find a link to register to the separate Zoom meeting. So follow the link, register, and you can follow it, and you can also follow it from YouTube if you don't want to use Zoom. Then tomorrow there will be the first of 3 roundtables on the pandemic, also with an interface with economics. We have a great set of panelists. And then on Thursday, we will have the last 3 lectures and 2 roundtables. So just to remind you what to expect from the roundtables, this is going to be a sort of free informal discussion between the panelists, and there will be time for you to ask questions, to express your opinion. So it aims at being an informal conversation. With that said, I'd like to introduce again Alvaro Sanchez, who is giving the second of 3 lectures on the assembly and evolution of microbial community. So thank you, Alvaro, for being with us. And when you're ready, you can share the screen and start the presentation. I think you're muted.
Am I muted? Can you hear me okay now?
Yes, perfect.
Perfect. Okay, so let me see if I can move this here. There you go. Okay, so I'm going to continue what I started yesterday and tell you about this work we're doing in our lab to try to understand the rules that govern the assembly of complex microbial communities using enrichment cultures. And I just wanted to, before I get into today's material, I wanted to just give you a brief summary of yesterday's most salient points, just to emphasize what the basic results are and what it is that we're trying to explain. So a big question that I'm very, very fascinated with is this idea of how reproducible microbial community assembly is. And this is a question that can be explored in natural systems. And in fact, it has been explored in natural systems. I give you an example of this one among many, but this one is particularly interesting, I believe. It's a work by Stylianos Louka and Michael Dobley and other collaborators, where they were examining the assembly of microbial communities in these water tanks that form within the base of the foliage of bromelia plants. And again, these are tropical plants. I think this particular study was done in Brazil, looking at water tanks in plants that are in close proximity to one another, so that many of the physical components of the environment are shared among those habitats: temperature, humidity, etc., and also the same regional pool of species. So when these folks looked at the microbiome of these different plants, they found that they did not contain that they were very variable, right, from plant to plant, and most of the plants, most of the OTUs were absent from each other, and only about 1% or less of all the OTUs were shared among all plants. So there was a lot of variation at the species level in these microbiomes. Yet when they looked at the metagenome and they examined the abundance of different genes involved in non-metabolic pathways, they found that the fraction of the metagenome devoted, for instance, to fermentation, to respiration, to carbon fixation, nitrogen respiration, and so on and so forth, those were very consistent from plant to plant. Right? And they found very similar quantitative ratios of these metabolic functions in all of these habitats, despite the fact that they find substantial taxonomic variation across them too. And this, I also mentioned that this same finding has been made in a wide range of other habitats. It's been made in marine environments, in the human microbiome, in algal communities, and the list is long. And however, it is difficult in natural habitats to disentangle what are the mechanisms that are underlying this phenomenon. There's many ecological forces that shape the assembly of microbial communities. Some of them are governed by chance, such as mutations, the arrival of species into a habitat, or dispersal. There's other ecological processes that are more deterministic, such as selection, which you would expect to be a force that will generate more homogeneity in community composition across habitats that are, at least those that are very similar. And finally, you can also have the confluence of the two, which we can call historical contingency, and where, which is, there's many different mechanisms that can lead to historical contingency. One of them is environmental modification, which is that when you have habitats that are very similar to one another, once they get colonized by bacteria, the metabolic activity of those bacteria will change the habitats in subtly different ways, and that will cause an inevitable level of historical contingency, because when you have different habitats getting colonized by different bacteria by random dispersal, those habitats that were originally very similar will become less and less similar as a function of time. So what we have next is that the problem to understand this question of reproducibility is that, in addition to being many different ecological forces that shape the assembly of microbial communities, we also have that the selective pressures that are present in most natural habitats are very difficult to know, right? We may infer them, or we might try to guess what the selective pressure might be through data or through just knowledge of the environment. But there are many different, if you think about it, right, we really don't know all of the physical and chemical and nutritional niches that might be occurring in particularly because they're so small, right? Many of these are molecular. and really understanding in detail what are the forces that shape selection in different habitats is something that is very, very difficult to do in nature. So the approach that we follow in my lab is we're trying to ask if it is possible to study the process of microcommunity assembly in synthetic habitats where we can know the selective pressures, we can know the chemical composition and the nutrient composition of these habitats. We know exactly what niches are we supplying, right? And we can also know for a fact what is all of the spatial determinants of microcommunity assembly. We can fix them. We can know the temperature, the pH, and we can control all of these factors. And we can also control ecological factors and ecological processes that contribute to microcommunity assembly. We can control the rate of migration from the regional pool. We can control what is the composition of the regional pool. We can control to some extent the population size, the connectivity between habitats, and so on and so forth. So the question we're asking is, if we know all of the things that we do not know or are very difficult to know in natural habitats, can we then understand more mechanistically the origins for these patterns of convergence at the functional level, but divergence at the taxonomic level that are found across a large number of natural habitats, right? So that's what my lab is trying to do. And in other words, we're trying to more mechanistically understand the reproducibility of microcommunity assembly. I gave you also an idea of what are the experiments and the experimental pipeline we're following in my lab. We're using high throughput enrichment communities. We take, for that purpose, samples from the environment. For instance, we take soil samples. We stick that in a bottle with a water saline solution. We filter this to eliminate all of the larger particles, and we're left only with the bacteria. And then we sample from that large bottle of bacteria into a small miniature test tube, and with a defined synthetic medium. And here we are supplying the nutrients to the bacteria that we have a lot of control over. In today's talk, those nutrients and the first part is going to be glucose. Later on, I'm gonna tell you about the results we get when we use other resources as well. And what we do is that after we inoculate by random sampling from the regional pool, we let the bacteria grow for a period of typically 48 hours. And after that period of growth, we apply a bottleneck. We take a small sample from here and add it to a new little test tube where we replenish the nutrients. We let that grow again, and we keep iterating this process. And I showed you, at the end of every 48-hour period, we are doing 16S sequencing to quantify the composition of different species in our communities. And I showed you also the results of a typical experiment. This is the relative abundance of different genera in one of these enrichment communities as a function of time, as a function of the transfer that we're looking into. This is with relatively shallow sampling, where if you think about it, we're just kind of sampling 10,000 random individuals from each community and identifying them, right? So that's essentially what we're doing. And the different colors here, each color represents a different genus, and the width of this column represents the relative abundance of that genus in the community. As you can see in this plot, after about maybe 8 to 9 transfers, community composition stabilizes and becomes quite constant through time. So what we are asking here is how reproducible community assembly is. And then the main question is, if we do the same experiment multiple times, do we find identical outcomes? That is the ultimate question we're asking. So to answer it, what we have done is that we have inoculated multiple replicate communities from the same regional species pool. And we can take, say, 8 different test tubes. We fill them all with the same medium, right? We put them in the same incubator, and we inoculate them from the same species pool. And what we find is that after we propagate those 8 communities in identical environments for about 80 generations or so, 12 transfers, and we examine the composition of these communities, we have found 2 things. So one is that we see very strong convergence at the family level, right? All of these wells contain essentially this, very similar ratios of the 2 main dominant families, but very much more variable community assembly when you look at it at taxonomic level of genus or lower, right? So this is reminiscent to what we have observed before in these studies that was mentioned before, right? There's a lot of variation at the species level, but a lot less variation and much more convergence at the level of family. Well, what they were observing actually is no families, is function, right? So our idea is that perhaps the two are connected, right? That this family level convergence that we're observing is reflecting a convergence at the functional level too. And just to give you an idea of what this looks like when we repeated our experiment with 12 different regional pool species, what we find is that this is across 12 different inocula, 8 replicates for each, and I'm plotting the family level composition of the communities for all of those experiments. What you can find is that there's a very strong reproducibility. Most of them are dominated by the same 2 dominant families. In blue, it's Enterobacteriaceae. In red, Pseudomonadeaceae. You have other more rare families that appear in some but not other of the communities. And even the quantitative ratios of these two are quite similar. I just wanted to give you another example of this kind of phenomenon of family level convergence. And this is work by my colleagues at the Connecticut Agricultural Station, Steve Blair and transects. And when you find in the study, what they did is they looked at community assembly in the stigma of the flowers of the apple tree. And they looked at the communities that assembled on about 100 different flowers. And one of the things they found is that, again, they find fairly consistent community assembly in these habitats at the family level of taxonomy, with Enterobacteriaceae again in blue and Pseudomonadeaceae in red, which are found across all of these flower communities. Yet they find a lot of variation at the species level. If you look at the different OTUs with the Pseudomonadeaceae and the OTUs within Enterobacteriaceae, you find that even though at the family level these communities are very similar from one another, they contain different specific members of each family, right? So this result again is consistent with this pattern. But in this case, again, they're finding this pattern not at the level of function, but at the level of family, which is what we did. And I'm just plotting these two things side by side so you can get an idea of the results are. I mean, of course, they're not identical, but these environments are very different, right? We're talking about a flower and a sugar-based synthetic medium. It's interesting because we think that the environment that these microbes are experiencing in the flower is, at least the nutritional environment, is not that different from what we find in our experiments. Anyway, well, this is all is painting a picture that presents a number of questions. So the first one is, why do so many species coexist on a single limiting resource? And this was the subject of yesterday's this lecture. Second is what we need to understand is, why is community assembly so convergent at the family level? Again, in our experiments, we're observing family level convergence. In others, they found functional level convergence, right? So we need to try to understand that better mechanistically. And in tomorrow's lecture, I will be talking about why community assembly is so variable at the species or genus level. And again, this was yesterday's, and that's going to be today. Okay, so today's what I wanted to go through is the work we've tried to do. I try to understand why we're finding these results. Why do we see that community assembly is so convergent at the family level? And matching this with the results that I've been hammering to you yesterday and today, our first guess is that this is actually reflecting some form of functional convergence. Right? And to test that idea, what we did is very simple. We took 13 or 14 of these, I can't remember the exact number of the communities that we had. I had shown you before that we had assembled in glucose as the only carbon source, right? So we took those communities and we did dilutions, and we spread them over agarose petri dishes, and we spread them at very low densities of cells, so that we could when cells would grow on these petri dishes, they would form little colonies, and cells from different species form colonies that are different from one another, right? So we could take those colonies, then we sequence their ribosomal DNA to to understand who they were and what was their identity, and compare those with the 16S ribosomal RNA sequence that we had previously obtained from community level 16S sequencing. And the two matched very nicely, so we were able to isolate a large number of the members of 13 different communities for a total of around 100 different Pseudomonadesia and Enterobacteriaceae strains, right? So we were able to take the members of those communities and separate them, right, and grow them in isolation from the community where they were isolated. And what we did then is that the first thing we wanted to do is we wanted to ask whether the Pseudomonadesia and Enterobacteriaceae, which is these two families, would differ at the family level in the growth rate in glucose. I remind you that glucose is the only carbon source we're supplying, and that Enterobacteriaceae is about 2 to 3 times more abundant than Pseudomonadesia in our communities. So we wondered if that was because Enterobacteriaceae is actually better at growing in glucose than Pseudomonadesia is. And so what we did is that we took each one of those isolates and we grew them on media containing, that is exactly the same medium where our community had been assembled. It is minimal media. For those of you who are knowledgeable about this is M9 medium with glucose as the only carbon source. And we determined the growth rate by just looking at the growth curve over a period of about 48 hours. We determined the maximum growth rate of the Enterobacteriaceae strains and the Pseudomonadesia strains individually, and we're plotting them here, right? And in blue, I'm showing the growth rate of the Enterobacteriaceae. In purple, I'm showing the growth rates of the Pseudomonadesia. And as you can see, the Enterobacteriaceae grows better and reaches higher growth rates than the Pseudomonadesia as a family. So what we find is that at the family level, there's conservation in the growth rate of in the supplied resource, right? Which may explain why Enterobacteriaceae is observed at higher abundance than Pseudomonadesia. The next thing we did, though, is I told you yesterday that an important component of coexistence in our communities is metabolic cross-feeding, that the Enterobacteriaceae, when they grow in glucose, they are known to secrete various metabolic byproducts, including acetate, succinate, lactate, as well as others. And these three are, we used mass spectrometry to determine what are the most abundant byproducts. And these three are the ones that came out as being the dominant ones. And one of the things that is interesting is that we find that the Pseudomonadesia here I'm showing is the amount of acetate produced over a period of 48 hours as a function of time for our collection of isolates. And in blue, I'm plotting those results for the Enterobacteriaceae, and in purple for the Pseudomonadesia. And you can see that as the Enterobacteriaceae grows on glucose, the amount of acetate they produce, particularly after 16 hours, and this is the relevant time scale, and I'll tell you why in a minute, is very conserved, right? All of these bacteria produce very similar amounts of acetate in these habitats. Now, the reason why 16 hours is the most important time scale is because this is the time that it takes for glucose to be exhausted, right? So what happens between this and that is that the single strains that are growing in isolation ended up then metabolizing some of their secretions. And in many cases, we find, for instance, for Citrobacter, which is these guys that you see over here, that acetate secretion continues for a little bit longer. And that's probably a byproduct of organic acid metabolism because the glucose has been exhausted by that time. But so at the relevant time scale, which is what happens with the glucose when they consume it, is that all of these Enterobacteriaceae produce very similar amounts of acetate, right, which is the dominant metabolic byproduct. They also produce similar, though less so, amounts of succinate and lactate. And what you can see is that there's a conservation of quantitative niche construction for all of these species of Enterobacteriaceae over with respect to by comparison with the Pseudomonads. And what's more is that we find that when you plot the amount of acid that is being produced as a function of the maximum growth rate that these bacteria can attain, you find that there's a fairly strong correlation between the two. And this is something that has been documented before for other Enterobacteriaceae, such as E. coli and Salmonella, which is that the faster they grow, the more acetate they release, right? And that is because more and more of their metabolism is shifted to fermentation rather than respiration, even though it's really called overflow metabolism. But more of their metabolism kind of stops at acetyl-CoA and then leads to pyruvate fermentation and secretion of acetate, and less of the glucose flow is directed to TCA cycle and to full respiration. So what that leads is that the faster bacterium needs to grow, the more overflow will have to do. And that leads to a strengthened secretion of acetate and other organic acids, the faster these bacteria grow. And that is also observed. Each point represents a different strain. This is for many different genera of the Enterobacteriaceae, including Rhabutella, Citrobacter, Enterobacter, Klebsiella, Serratia, and others. And you can see that this kind of trade-off between yield and growth rate is applicable to broadly to all of the strains of Enterobacteriaceae, regardless of the specific species or genus they are. By contrast, Pseudomonas doesn't engage with that, as is known, right? That overflow acetate production by overflow metabolism is not documented to occur in Pseudomonadesia, which is primarily a respiratory bacterium. Okay, so it is believed that this trade-off emerges from physiological constraints in protein allocation, but that's a subject that is beyond the scope of this talk. But the point I wanted to make with this slide is to illustrate that this emerging picture from the two slides I've shown you before. First is that the glucose is selecting for fast growers and strong growers on glucose. And the faster, and we find that Enterobacteriaceae grow faster than Pseudomonadesia. And we find that the faster these cells grow, the more acetate they produce, right? So strong selection for fast glucose growers leads to bacteria secrete organic acids, and which do so, the amount of secretion are relatively similar. Again, the variation you see here on the y-axis is not that high, right? It's around 10 plus minus 5. But also we find that leads to the secretion of organic acids in a manner that is correlated with the amount of growth you have. And the next thing we did is we repeated those first measurements of growth rate for all of the other dominant fermentation byproducts that are released by the Enterobacteriaceae, acetate, succinate, and lactate. And what we find is that in this case, the Enterobacteriaceae grows on average less well on those byproducts than the Pseudomonadesia does, right? And so we find that both for acetate, succinate, and lactate, the Pseudomonas grows faster on average than the Enterobacteriaceae. So we also find that in addition to there being phylogenetically conserved growth of the bacteria in the supplied resource, where Enterobacteriaceae grows better than the Pseudomonadesia in glucose, there's a convergence in niche construction, and there's also a convergence at the level of growth in the byproducts of glucose metabolism, where in this case the Pseudomonadesia grows better than the Enterobacteriaceae. And I just wanted to show you this. This is further evidence for this thing I'm trying to tell you. Let me see if I can move this here. What we find here is I'm showing for a specific community the concentration of glucose as a function of time, and the ratio between Pseudomonas and Enterobacteria. And this RNF ratio I will explain in a minute, but this is the ratio of Pseudomonas to Enterobacteria, right? So we see that glucose is depleted, and after 21 hours, there's none. Actually, it's a little bit earlier than that, but in this plot, there's what we quantified. And acetate goes up as glucose is being depleted, peaks at around 21 hours, and then drops to 0 after 48 hours for this one community. And at the same time, we find that the ratio between Pseudomonas and Enterobacteria declines in this first phase when glucose is being depleted. And this is consistent with the idea that the Enterobacteriaceae are consuming the glucose, right? And after in the last 23 hours or so, 27 hours or so, you find that as the acetate is being depleted, the ratio of Pseudomonas to Enterobacteria in this community goes up, right? And that is consistent again with the idea that the Pseudomonas are eating these organic acids primarily. Again, this doesn't mean that none of the glucose is being eaten by the Pseudomonas or none of the organic acids are eaten by the Enterobacteriaceae, We have every reason to believe that actually both of those things are happening. But the primary consumers for the glucose is the Enterobacteriaceae, and the primary consumers for the organic acids are the Pseudomonadeaceae. We've done other experiments that confirm this point. We have truncated the growth time at 24 hours, and when you do that, and the Pseudomonadeaceae goes away, and the community becomes entirely dominated by the Enterobacteriaceae, and with organic acids that accumulate and no one eats them. Okay? So we have substantial evidence that that's what's happening in our communities. Let me see if I can put this back in here. All right, so let me give you a summary of what we think is happening in our at the functional level. We have these communities that assemble into very similar ratios of these two dominant families, Enterobacteriaceae in blue and Pseudomonadeaceae in red. And what's happening is that the Enterobacteriaceae are being selected for by the glucose because they grow faster. And as one thing we observe is that the faster they grow, the more organic acids they produce. So as these bacteria and Enterobacteriaceae are growing on the glucose, they are releasing organic acids like acetate, succin, and lactate. And as they accumulate, the environment in the second half of the incubation time of this 48-hour period, the environment is no longer a glucose environment. It's primarily an organic acid environment. And in those environments, the Pseudomonadeaceae has an advantage, right? And that's what we're seeing it here. So the Enterobacteriaceae are occupying a functional niche, which is that of respirofermentative bacteria that specialize on the glucose, whereas the Pseudomonadeaceae are occupying a functional niche, which is that of respirative bacteria that specialize on organic acids. And we call this respirative functional group R and respirofermentative group F. And the coexistence between these two groups is stabilized by a metabolic cross-feeding that primarily goes from one direction to the other. Although that's only the first order effect. There's also, and we have evidence for that too, all other byproducts that are being released by both, right? So it's not that we're saying that the only thing that happens is that the glucose goes entirely to one and not the other. We know that is not true. We're just describing a first order effect and what is the primary consumer of each of the two niches. All right, so all of this, what shows to you is that this family level convergence we have observed and does represent functional convergence, right? And it is so through the evolutionary conservation of the relevant functions for growth and fitness in our environments. But now the next question we wanted to ask is whether it is possible to take this ratio between R respirators and F fermenters that we're seeing here and explain that quantitatively from the known physiological and biochemical processes that are occurring at the cellular level. So again, we get a ratio between Pseudomonas and Enterobacteria that is around 0.27, and that is the ratio between respirators to fermenters in our ecosystems. And to see if it's possible to recapitulate that finding, what we have been doing is genome scale metabolic models that are based on flux balance analysis. And I'm not going to go into details of how FBA works, but suffice it to say, this is a genome scale metabolic model where you could take a metabolic network. You can take, for instance, a metabolic network of a bacterium like E. coli, or you could make, as we did for this particular project, we built a super metabolic network that contains all of the known metabolic reactions in prokaryotes and put them together into a big matrix. And what you do then is you give that network an input, which is a set of nutrients. And with flux balance analysis, what you do is you calculate the vector of metabolic fluxes that would optimize growth. This is optimized a given biomass function that you give the model in that environment that you're supplying. And what FBA will do is will find what is the vector fluxes that will maximize growth. And that vector fluxes will give you an output, which is the amount of biomass produced per unit molecule consumed. You could put it that way. And also an output, which is the byproducts that are released in the process of growing optimally on that substrate. So what we did then is we looked at we took two models, one of an Enterobacteriaceae, an E. coli, and another one of I think this is wrong, this is actually P. putida, It's another of a pseudomonas, so it's in this case P-Putida. And what we did is very simple. We took and calculated what it would be per glucose molecule, well, how much biomass of E. coli would be produced, right? And how much acetate would be secreted. And now we took the acetate that had been secreted and fed it to this model of P-Putida and calculated how much biomass will P-Putida produce. And this was by taking basically these off-the-shelf models and evaluating their growth. And what we find with E. coli and P-Putida is the predicted ratio of P-Putida to E. coli biomass per glucose molecule that enters this trophic chain that we created was actually around 0.3. Again, this is only true if all of the glucose goes to E. coli and all of the acetate goes to P-Putida, right? But even with these very simple assumptions, we get a ratio between P-Putida and E. coli that is very similar to what we found before, which is around as being the average ratio of respirers to fermenter, which is 0.27. And we repeat the same exercise for a large number of previously well-curated models, metabolic models for both Enterobacteriaceae and Pseudomonadacea. And for each of those models, we calculated we did exactly the same exercise that I showed you before. And we calculated the ratio of biomass, of the Pseudomonas biomass to Enterobacteria biomass. And here we're plotting every possible pair of those 100 models. And you find that actually that value of 0.3 that we just found before is not an outlier. It sits very, very close to the average ratio that we find between Pseudomonas and Enterobacteriaceae biomass. And by comparison, we show you here the ratio of Pseudomonas to Enterobacteriaceae in all of our experiments. And the two are fairly close, right? This is really not a prediction per se. What we're trying to argue here is that one can explain these ratios from very simple arguments. And assuming that, making the simplifying assumption that all of the glucose goes to Enterobacteria and all of the byproducts go to Pseudomonadacea. And when you do that simple exercise, what you get is something that seems very similar to what we find in our experiments. Right, okay, so now the last thing I wanted to show you is results that we've collected a while ago, and this paper is now being written at the moment, and where we did the same experiments, but on a large collection of other sugars. And here the question was, okay, we found that this ratio of around 0.3 of Pseudomonas to Enterobacteria, or respirers to fermenters, and we found it for glucose, right? But how different would it be if we had used other sugars? What if we had used, I don't know, galactose, which is another hexose, or ribose, which is another sugar, in this case a pentose, or any other sugar alcohol like inositol, mannitol, or glycerol, right? So we did that experiment. We repeated this enrichment experiment that I've been discussing before, using two different inocula. These were two different potted plants in Josh's house. And from that soil, what we did is we established communities on environments that contain either one of these sugars in isolation. Just again, not all of them this mixed, but one at a time, right? And here on the x-axis, I'm plotting the identity of the sugar we're adding in. On the y-axis, I'm going to plot the ratio between respirers and fermentative bacteria. This dashed line marks the average of the glucose communities that we had seen we have been showing you before, and this gray zone here is 95% dispersal around that mean for the data. Right. So when we plot this data on this plot, what you find is that for all the sugars, we find a very similar metabolic structure. Right. The ratio of respirers to fermenters are extremely close to the value that we found for glucose. There are some outliers, but by and large, the results are very consistent. And just as a control, we wanted to see if, okay, what if we don't use a sugar, right? What if we use a metabolite, a nutrient that cannot be easily fermented and would be more likely to be respired? And to that end, we used a collection of different organic acids many of which are fermentation byproducts, but others are just, components of the TCA cycle. We also used some organic alcohols and a bunch of other nutrients, and repeated the same experiment from the same two inocula, but now we assemble them on each one of these collection of nutrients. And we find is that the ratio of respirers to fermenters when you do not use sugars is very different from what you get when you do use sugars. And this experiment is quite interesting. For instance, I think I particularly like this data. This here is pyruvate, which is an organic acid that sits in between glycolysis and the TCA cycle, right? And it's kind of intriguing that you get something that is kind of a transition in between the kind of ratio between respirers and fermenters that you see in the sugars and this cloud that you see here when you use organic acids. Okay, so I guess this plot would make the case that when you have similar nutrients, you are expecting to see similar community compositions. But can we more quantitatively define what nutrient similarity is? I mean, we're saying that sugars are similar and organic acids are similar to one another, but I'm basically waving my hand so far, right? Can we make a more precise definition of how similar to different nutrients are? So we again resorted to flux balance analysis, and what we're doing here is very straightforward. We're taking again our metabolic model, and we're feeding it different nutrients, right? So for instance, we're feeding one nutrient, and we're calculating the vector of metabolic fluxes, and we can plot this vector in that space of metabolic fluxes. And now we could take and through the same metabolic network, I mean, we can, in this case, what we're doing is we're constructing a universal metabolic network that contains all of the biochemical reactions, metabolic reactions that are known. And we're asking what would be the optimal way to metabolize each nutrient, right? So this is the vector of metabolic fluxes for nutrient A. This is a vector of metabolic fluxes that would be optimal for another nutrient B. And the distance between the two can quantify how different those two are, right? So if two nutrients are very similar, then they're going to be metabolized optimally in very similar manners, right? And if they're very different, they're going to metabolize in very different manners, right? For instance, glucose and galactose are metabolized very similarly, and they're going to give you a very small distance between the two, whereas glucose and, leucine are going to be metabolized more differently, and they're going to give you a larger vector here, a vector with larger distance. Right. So the next thing we did is we took this library of carbon sources that we had studied experimentally, and then we calculated the metabolic distance between all of them. And the first thing and then we did simple hierarchical clustering. And one of the things that was very reassuring is that the results we got from this exercise made a lot of sense. We find in this dendrogram that all the sugars are clustered together. All the carboxylic acids and organic alcohols are also clustered together. We also find that within these two groups, their structure, here you find in this neighborhood around here, most of the hexoses and all this glucose-containing disaccharides. And here you find most of the sugar alcohols and the pentoses. And here's the same thing, right? In this cyan group over here, you find all of the TCA cycle intermediates. These are organic alcohols and so on, right? So that there is within this, the structure that makes sense, right? That one would expect perhaps that those nutrients would be more similar to one another or less. Right, so then what we ask is to what extent this similarity between carbon sources can predict quantitatively similarity in community assembly in those nutrients. And we find is that it does actually do a very good job. So here I'm showing you the composition of at the family level for all of the sugar and sugar alcohols. Here now in blue, you find the Enterobacteriaceae, and in green, the Pseudomonadaceae. This orange guy here is Alkaligenaceae. And if you compare that with the organic acids and the alcohols, you find that it's quite different. In most cases, the Enterobacteriaceae is gone, and these are dominated by respiratory bacteria like Pseudomonadaceae, Alkaligenaceae, and others. But even within the sugars, the structure, in this first group that are made by glucose-like sugars, we find only very rarely that you find Alkaligenaceae. Whereas in this other group of pentoses and sugar alcohols, you find that Alkaligenaceae is much more common. And in fact, it's interesting that here in galactose, when it isn't, actually this is probably just a misclassification because of the nature of the hierarchical clustering algorithm, because the galactose is a hexose. The same is true for the organic acids and the alcohols. If you look at the TCA cycle intermediates, you find that all of them contain enterobacteriaceae here in blue. And even within those, there's even structure. So this group here is formed by fumarate, malate, and succinate, that they enter side by side in the TCA cycle. And all these 3 carbon sources recruit rhizobiasia, and they are the only ones that do, right? It's an endemic species for this group of carbon sources. Likewise, there's endemic species in this group here. Sphingomonadesia is not found anywhere else. And also there's convergent community composition within this other group as well. So now the question is whether the distance between nutrients can explain distance between composition at the family level. And what we decided to do is just plot one against the other, right? We can, at the family level, you can also have the communities assembled in nutrient A and the communities assembled in nutrient B, and you can calculate the family level composition for both. And once you have both, you can calculate the distance between them, right? And when we did that, we were plotting here the Euclidean distance in metabolic fluxes against the Euclidean distance in community composition. And what we find is that there is a fairly decent correlation between the two. And this is just a very crude measurement. I mean, we're now trying to extract, do smarter ways to extract information than simply plotting the entire distance of the entire metabolic space. Maybe there are some components that are more telling of how different two nutrients are, than simply the entire distance of metabolic fluxes, and to see if there's more signal that can be extracted this way, but this is where we are at the moment. We've been also using machine learning algorithms to see if it is possible to predict what's going to be the communities assembled in a new carbon source from a new inoculum. So just very basic, doing cross validation at this moment. And what we find is that for the families that are most represented, like Enterobacteriaceae and Pseudomonadesia, there's a very decent, the very simple machine learning model is capable of predicting the family level composition. And the same is true for function, which is even better, right? So if you now group the taxonomic composition by whether they are fermentative or respirative bacteria, and again, you train a model with one inoculum and with a set of carbon sources, and then you investigate what would be the expected community composition from another inoculum in one of the carbon sources that you left out of the training set. What we find is that it is possible to measure fermenter. We get results that make sense, right? And that the model is predicted to some degree. And I mean, this is all very preliminary work, but I just wanted to give you a sense of where we're going with all of this. All right, so what I wanted to tell you today is what we think are the reasons behind the fact that community assembly is so convergent at the family level. And what we had guessed that this could reflect a functional convergence because people had seen metagenomic convergence when binned by metabolic function, right? So our original guess was that the family level was representing the evolutionary conservation of metabolic traits that are functionally important in our ecosystems, and that's why we're seeing the signal at the family level. But because our environments are so simple, one is capable of resolving these questions mechanistically, and that's what we've been trying to do. And we are finding that when you have sugars, and not only glucose but any other sugar, you find that there is a very predictable ratio of bacteria that are respiro-fermentative, that are found, that are specialized in the sugars, and there's another group that are respirative that specialize in consuming the byproducts released by the former. So one of the things that is interesting about this result is that when we tend to think of metabolic traits, in particular the consumption of carbon source of nutrients that are based on carbon, the previous work has found, right, that this is a highly variable trait. It's a trait that is not supposed to be conserved at the family level, right? So if you look at, for instance, by contrast, there's other metabolic traits like the usage of specific electron acceptors that are much more deeply conserved evolutionarily. And I just want to give you one example, right? This is an experiment that was done with the bacterium E. coli that was grown in, I'm sorry, I'm missing the reference here for some reason, that it was grown in a collection of different nutrients, and it was assessed whether E. coli could grow or not, right? And these authors took I think it was 150 different strains of E. coli and close to 100 different nutrients. And they're just measuring whether growth or not growth on each one of those. I'm just zooming in because this plot is a bit of a mess. But here you have, for instance, a subset of these strains, and you have serine, raffinose, and sucrose, which are 3 different nutrients. And as you can see, some of those strains can grow on serine, but others cannot. And these can, the others cannot, right? The same thing is true for raffinose, sucrose, and most of these other carbon sources on this group here. Only 7 of the carbon sources tested were able to be used by all of the E. coli. Okay, so what this suggests is that metabolic traits are typically thought of as being shallow, right? And that it's easy for bacteria to gain or lose the ability to metabolize a specific substrate. So that's kind of seems to be contradicting what we found, right? That there's family level conservation of these metabolic traits. But I just wanted to bring up again that what we've been studying is not the ability of a microbe to use a substrate or not use it, but rather how good that microbe is at using it, right? We're measuring quantitatively how much how well a bacterium can grow in a given substrate, and what byproducts are being released, and how well those other bacteria can grow on those byproducts. It's not just that you can use glucose. In fact, we find that glucose can be used by both Enterobacteriaceae and Pseudomonadeaceae. It is how well you can grow on those that determine whether you can be found in that environment or not. Because again, as you may also very intuitively think, just because a species exhibits a trait doesn't necessarily mean that that trait is going to be relevant for the ecological role that that species plays in nature, right? And that is true for bacteria as well. And I think this is making a case for us measuring quantitatively not just basically to measure the realized needs, right? And to quantify how competitive microbes are on different carbon sources if we want to really understand whether a microbe will be found or not in a given habitat. And that's it. Again, this work was done by the amazing people in the lab, and I think we may still have time for some questions, so shoot.
Great, thanks a lot, Alvaro. So we have time for questions. Yes, there is one from Silvia.
Yes, hi. I wanted to ask a very general question on the patterns that you showed also in the previous lecture and at the beginning, the fact that the functional composition and the family composition are constant. So I was wondering, can we compare these patterns with a null model that would tell us that we would expect more variability in the absence of a mechanism that brings this constants?
Right. Right. Yeah. The question is, what would be the null model, right? I mean, like, yeah. I mean, like, you may imagine multiple different null models for community assembly, right? You could have neutral models for community assembly. You could assume that all nutrients, that basically that all of these species are equally good at eating all the nutrients that we're providing them. So I guess, yes, I mean, it is completely possible to compare the expectation to null models, right? If you just sampled randomly, I guess the simplest one is that if you say that you sample randomly species from the regional pool and put them together, right? In our and the question you may ask is then, do you see the patterns that we observe? And the answer there is no, you don't, right? Like there is no family level conservation of any kind. And in fact, depending on what nutrient you add, you're going to find different species on each, right? So that already tells you that there is a very strong selection that is shaping the assembly of these communities. And that's if you compare this to very neutral models. Now, And then, of course, if you're trying to compare these patterns to other null models, I guess, like always with null models, right? The null model needs to reflect a null assumption or a null hypothesis, right? So so you would need to define what that null hypothesis is, and then absolutely you could create a null model that predicts what that expectation would be, and you could quantitatively compare findings with that, yeah, or the patterns we observe with that null model. But for the very simple ones, which you may imagine is just simple randomly drawing ESVs from the original pool and asking whether the same is true, the answer is it isn't, right? We have checked, and that is for sure not the case. But as for other null models, again, it would depend on what the null hypothesis that you're trying to disprove is.
Okay, thank you.
Great. There is then a question from Kiseok.
Hi, thank you for the great talk. And my question is, when you're doing the simulation with the FBA model and the experiments with this metabolic model for like Pseudomonas and Enterobartesiae, do you use like 100 strains for each of these family, or like how do you represent the family?
Right. So we took just models that were well benchmarked, that people had done a lot of work with before. And so I could send you the list of the models we used if you're curious. Some of them, they're basically published models by other people that, and then we had to just adjust them a little bit to the specific environment that we had. But this was not our models, right? We're taking models from other groups, and they were not 100. I think I can't really remember the actual number for each of the two. I think the total, it was around 70 something models, I believe, but they weren't equally represented. So I think there were more interactivities than Pseudomonads, in fact. And the Pseudomonads were not sampling the entire phylogeny of the Pseudomonadesia. So there's some degree of non-homogeneous sampling of the two families in terms of the models, right? We focused on those metabolic models that they were well benchmarked experimentally before, right? And if you want the list, I'm happy to send it over if you're interested.
Thanks. So for the experiments, you did use your like different kinds of strains for each family, right?
Right.
Okay.
Right, and also they're not, again, we were not trying to sample the entire phylogeny of the Pseudomonadesia and Enterobacteriaceae. We were already biased by those bacteria that came up in our communities, right? So we took bacteria that from communities that had assembled in glucose and both Pseudomonadesia and Enterobacteriaceae. And we also did similar experiments with random soil isolates that were not in our communities, and we included them into the set. And we've done another set of experiments. The results are not that different, right? So it doesn't seem that, I mean, there's some patterns, but it's not very obvious that the randomly selected bacteria from soil are very different in their patterns of secretions, or even maybe have a slightly lower growth rate than the ones that have been selected on average, but the patterns of secretions were similar, and we didn't really appreciate any significant difference. But we have not really tried to do a very exhaustive search for Enterobacteriaceae, Pseudomonadesia to see how conserved they are throughout the entire taxonomic group.
Okay, thank you very much.
Great. There is a question from Martina.
Hi, thank you. This was all very interesting. And I have actually two questions, if I can. One is, how do you, I was looking at your plots before, and how do you justify the fact that the Pseudomonads don't really have different growth rates in acetate? When you show the different plots.
Let me see if I can get your question right.
Yeah, sorry.
Here?
Yeah, for here. it seems that you have definitely two strains that grow much better, but they're not really different from one another, or am I getting it wrong?
Each of these is a different strain, right? So each of these is a different strain. The strains have different 16s, right? So we're not even, every single one of these has a unique 16s sequence, right? And we did Sanger sequence for the entire region. So it's a high quality sequence. So they are different, all of these strains, yes.
No, but my question is, it's not the big difference between Enterobacteria and Pseudomonads. And that's why I was wondering how then you justify these changes in acetate, even though the growth rate doesn't appear to be completely different. And while
but it's different, right? I mean, I think even if you remove those two strains, it's statistically significant, right? And this is just the maximum growth rate. We also find that if you look at the average growth rate, which is not just the maximum you have, but basically counting the lag phase and how long does it take for them to reach half of the maximum growth they have, Pseudomonas also grows better than Enterobacteria. And the same thing is true for lactate. I mean, even though we're focusing a lot on acetate, concentration of lactate actually is not that. Let me show you here. It's not that much lower, right, from what we see in acetate, and there's more carbon per. So lactate is more valuable than acetate metabolically, right? So we think it's actually something you cannot neglect, and we're now trying to understand exactly how it's not just the acetate itself; it's a combination of acetate, lactate, and succinate, and other organic acids that together recruit the Pseudomonadeaceae, right? So it's not just one, and it also is not just the growth rate, right? It's the growth rate is defined. I mean, as you know, the growth rate changes right over the entire growth period, and you have on the one hand the maximum growth rate, which is important, but of course that is only sustained for a brief period of time by the entire population, right? And how rapidly you get out of lag phase and the average growth rate over that period is also an important parameter. But just to clarify, this is statistical significance is not just because of these two guys, right? It's the entire distribution, and this is only reflecting the maximum growth rate. But the same patterns would be seen if we had. I was showing you the average growth rate over the entire over the first. I think until we trunc it until the first. I think it's T1 half, right? Until it reaches half of the maximum OD.
Okay, and if I can link this to another thing, so I was wondering if these two strains that have higher maximum growth rate are then those that are more abundant in your experiments. And related to this, I was wondering if when you find this convergence, whether it's driven by the most abundant strains, or do you think that this has nothing to do with abundance?
No, I mean, okay, so it's a good question. So I really don't think we have correlated the growth rates, and something that we've been meaning to do for a while, the growth rates of these bacteria in isolation with their abundance at the community level. This is an idea that we've been floating for a while, but haven't quite done it yet. Although we have all the data, it shouldn't be very difficult to do. The other thing that you brought up that I think is quite interesting is if you look at this data, right, and we see there's convergence, there's still a substantial variation, right, among the communities, right? There's still variation in the ratio between Pseudomonas and Enterobacteria. And I absolutely agree that, and in fact, we have some evidence that points to that, right, that which is the identity of the dominant bacterium impacts the actual RF ratio you see. I'm going to share some data tomorrow that will cement that point, that when we have alternative stable states from the same inoculum, we have looked at the specific ratio of respiratory to fermenter on each one of the two. And depending on which is the identity of a respirator, if it's a Pseudomonadeaceae or an Alcaligenaceae, they don't quite reach the same abundance. There's variation between the two. And I think because these communities are very small, right? So we're still talking about 5 to 20 species, but most of them are very rare. So there's still going to be a variation on which is exactly the Pseudomonas you find on each community, right? If these communities were larger, I think all of these things would average out, and you would probably have more convergence. That would be my guess, but we don't know, right? But because you only have 3 species, right? Which one happens to be the dominant member of its taxonomic group? Of course, adaptation to the specific dominant carbon source is not the only selective force we have, right? And any other differences, as well as variations on adaptation to a carbon source that we also find should matter for this RF ratio. So that's what we're focusing on, averages rather than the fluctuations, because this is an area we haven't yet explored. But I think you strike a very fair point that I think it's very likely that if you examine in more detail and try to understand the fluctuations around this average value, and that could be correlated with traits of the specific taxa that are found in those communities, right? And maybe there are correlations between them. We just haven't looked yet.
Okay, thank you very much.
You're welcome.
Great. I don't see any other questions in the chat or in the participant list. So if there are no other questions, well, thanks Alvaro again for the lecture of today. Just to remind that the next lecture by Alvaro is going to be in 2 days on Thursday. So thanks again. And now we are gonna split in the breakout rooms for 10 minutes, and we're gonna be back at 5 p.m. Italy time for the seminar by Alejandro Rodriguez Verdú. So thank you very much to everyone, and thanks Alvaro.
Bye.
Okay, welcome back to the main meeting room. So everyone should be back from the breakout rooms in a few seconds. In the meanwhile, if you are following from YouTube, I remind you that you can ask a question by posting them in the chat. While if you're following from Zoom, you already know that you can either post the question in the chat and I read it for you, or use the raise hand tool in Zoom. And if you just connected, I remind you to check the program, and that tomorrow at 4 p.m. there will be a colloquium by Ned Wingren, and you need a separate link to connect to that, but you'll find all the details and the registration link on the program of the school. So with that, it's my pleasure to introduce the next speaker, Alejandra Rodriguez Verdugo. Alejandra is a professor at the Department of Ecology and Evolutionary Biology at University of California, Irvine. And she works at the interface between ecology and evolutionary dynamics, doing experiment trying to combine the two. And today is talking about the evolution of microbial interactions in fluctuating environments. So thank you very much, Ale, for being with us, and feel free to share when you're ready.
Yeah. All right. Can you see my screen? Thumbs
Yes,
up.
works perfectly.
Excellent. Well, excellent. So thank you so much for the invitation. I'm super excited to be here and to talk about my work that I started at ETH Zurich when I was a postdoc with Martin Ackerman, and then I'm continuing this work right now in the Department of Ecology and Evolutionary Biology at UC Irvine in California, in the US. And today I would like to talk about evolution of microbial interaction in fluctuating environment. But first, why do we care about microbes? So microbes are the oldest organisms on Earth, and they have been able to colonize any possible environment on the planet, from the driest places, such as the Atacama Desert in Chile, to the icy lakes of Antarctica. So microbes have been able to thrive and adapt to the harshest condition. And not only they have amazing adaptability, but we know they are very important for life on Earth. So many of these microbes underlay the biogeochemical cycling on elements on Earth, and then therefore are essential for ecosystem functioning. And we know that many of these functions are not performed by microbes in isolation, but they are performed by many microbial species. That means it's really microbial communities that underlay these functions. And now we know also that these microbes are really relevant because, for example, they can influence climate change by mediating the carbon atmosphere land exchange, and therefore they can actually feed back into the climate. So it's very important that we understand how these microbial communities respond to change. Also, we know that these microbial communities live in close association with animals and plants, and therefore define their well-being and keep us healthy, but sometimes also cause disease. And here in this image, I'm showing a beautiful microbial community that lives in our gut, in the human gut. And this gut microbiota we know is very important for keeping us healthy. And there's recent studies that have shown that disrupting these communities with changes of diet or antibiotics can actually lead to chronic disease such as Crohn's disease or Alzheimer or anxiety. And even more recent studies can also show that they actually control cognition and behavior. So it's very important that we understand then how disturbing these microbial communities can actually influence our health. And then finally, microbes can help us. So they are used in industry, and for example, they are used for energy production, and they are the next generation of biofuel, which are clean and renewable. And also now in industry, they are engineering whole microbial communities, for example, to improve the health and the fitness of the host that carries them, for example, plants. So given their importance for the environment, for health, and for industry, more and more people are trying to understand how these microbial communities work. And although this seems a very simple question and very straightforward question, we still don't have a good answer for this. And the reason is because microbial communities, as you already know, are complex systems. So if we look at it, they are composed by populations of one single species, which are interacting with population of a different species. And then this interaction can actually lead to emergent properties that give certain functionality at the group level. And finally, we have to consider that many of these microbial communities live in structured environment and fluctuating environment. So then the environment also is not static and changes. So one way to approach then this complexity is what we can do is to just isolate some of these species, and instead of dealing with the whole complexity of thousands of species from nature, we can just assemble these few species in what we call synthetic communities, and then we can bring them to the lab and study them in the lab. So this is the approach that I'm following now in my research group. And then in my research group at UCI, we study two questions. Overall, we are interested in understanding how do species interaction influence evolutionary dynamics. And on the other hand, we're interested in understanding how changes in individuals can influence evolutionary dynamics. And in general, we follow a bottom-up approach, which probably Alvaro Sanchez talked a lot about this. So basically, we use this synthetic communities simplified system. And what we do is we try to build quantitative prediction with mathematical modeling that we can then test with experiments. And then these experiments can inform, can feed back to inform our quantitative prediction and mathematical models. And for today, I would like to talk about one project which look at how the species interaction influence evolutionary dynamics. And this is in the context of the ecology and evolution of pairwise, positive pairwise interactions. So this work, again, was done at ETH Zurich in collaboration with Martin Ackermann. So what are positive pairwise interactions? So positive pairwise interactions are any interaction between two species where either one of the species get a benefit from the interaction or both of the species get a benefit from the interaction. And we know that these ones are very important for ecosystem functioning. So for example, we know this kind of association occur in nature. And for example, we can think about this mutualistic association between methane oxidizing archaea and sulfite reducing bacteria. And these ones are very important for deep subsurface ecology, and they are really the base of these ecosystems. But for example, a more familiar example, you might be more familiar. So coral reefs, for example, is this association between an animal and an algae. And we know that this actually positive mutualistic association is really the base for these shallow marine ecosystems. So therefore, given the importance for the environment, more people are trying to understand how stable are these positive interactions. But then one thing one has to consider is that the environment is not static. And for example, we can have that there's changes in resources, and these changes in resources in the environment can alter species interaction. So very simplistic, one can imagine the interaction between two species, a squirrel and a bird, that live in an environment with a lot of resources and different kind of resources. So we have that they have a neutral interaction. But then if the condition changes, and now we have that actually they are in an environment with limited resources, now we have that this neutral interaction changed to a negative interaction, and now they are in competition. So then we should try to address this question of how stable are positive pairwise interaction, considering that the environment also varies. So to address this question, I use this synthetic community composed by these two bacterial species, which is Acinetobacter and Pseudomonas. And these two bacteria are very interesting because they have amazing metabolic capabilities. Also, they were isolated from nature, from a polluted aquifer in Denmark. So they coexist in nature. And what we did was to just isolate them and bring them to the lab. And then the metabolic capabilities that they have is that they can degrade aromatic compounds, which are very hard to degrade by many other microbial species or the other species. So they definitely are of interest for bioremediation. And we have that, for example, we can tune the interaction depending on the resources. So for example, if we grow these two bacteria in an environment where we supply them with benzyl alcohol as a carbon source and carbon of energy source, we have that only these bacteria, Acinetobacter, can use these benzyl alcohol. But then if we grow them in the presence of these other bacteria, what happens is that Acinetobacter can actually oxidize this benzyl alcohol, and then it accumulates an intermediate product, which is benzoate, which passively leaks out in the external environment, and then in the external environment, Pseudomonas can utilize this benzoate. So we have, in ecological terms, a commensal interaction where Pseudomonas get benefited by the presence of Acinetobacter, and Acinetobacter is neutral to the presence of Pseudomonas. So we have this cross-feeding interaction. But then now if we change them in a minimal media where we grow them with citrate as the limiting nutrients, we have that these two species compete for this citrate. So now we have a negative interaction. So as you can see, it's very nice model system because we can then tune interaction depending just on the carbon source. So with this model, so first, how do we measure interactions in the lab? So the way we do it is we use a batch culture system. So that means we just grow these bacteria in a shaking tube with these nutrients, and then we wait until they grow 24 hours. So at the beginning, they have a lag phase, then they have exponential phase, and then they saturate the nutrients in the media. And then after this whole cycle, after 24 hours, we transfer 1% of the population into a fresh medium, and we continue these cycles for many days. So at the end, the kind of results I'm going to be showing is this kind of graph, where in the x-axis, I have the time in days, and in the y-axis, I have the cell density, which we estimate as colony forming units in agar plates. And then in this particular example, for example, you can see that the blue type is able to sustain this serial dilution regime. So we say that it has a stable population over time. While, for example, in this particular case, the red type, even though it's able to grow, is not able to sustain this serial dilution regime, and it gets extinct after 4 days. So the first thing we wanted to see is to do these experiments first in what we do is monoculture. So that means we grow them in isolation to see how these bacteria behave. And we can see that Acinetobacter is very good at using this benzyl alcohol. It achieves high cell densities, while Pseudomonas is not very good at utilizing this benzyl alcohol. But then when we do these experiments in co-culture, so now growing them together, we have now a different situation where we have that Pseudomonas achieves 2 orders of magnitude higher cell densities in the presence of Acinetobacter than just alone. And then what happened in, so also we can visualize actually these kind of interactions using microfluidics and coupling with a microscope. So what I'm going to play now, just to visualize this direct cross-feeding interaction, is I'm representing Acinetobacter as these round cells, and then Pseudomonas is in green, like a road shape. So you can see that actually, if we play this time-lapse movie, we have first the growth of Acinetobacter, and this growth is followed by this kind of green fluorescence, and that's the growth of Pseudomonas. So we really see this sequential growth where we visualize this cross-feeding interaction. So now, what in the other condition, so in the condition of citrate, when we do our experiments in monocultures, growing them in isolation, we have that both species can grow well in monoculture and achieve high densities. But now when we put them in co-cultures, we do see that actually Pseudomonas is a very good competitor, and is such good competitor, and it utilizes this citrate so well that it actually outcompetes Acinetobacter only after 5 days of growing them together. So we can see this very asymmetric competition situation. So what we wanted to test in this experiment is then how are these, I mean, can we predict this ecology and evolution of this pairwise interaction? So what we did was to ask two questions. So how stable are these positive pairwise interaction in fluctuating environment? So not just keeping the environment constant. And in particular, we asked what happened if we interrupt this positive interaction with period of competition or negative interaction. And our second question we address is how stable are positive pairwise interaction over evolutionary timescales, both in constant and in fluctuating environments. So for our first question, the first thing we did was to build a mathematical model, and this was done in collaboration with Clément Villain, which is right now at the University of Zurich. And what we did was to build this mathematical model that we parameterized with single species growth measurement. So for this part, what we did was to just do growth curves assay of these monocultures. And then from there, we also estimated, for example, the excretion rate of this benzoate by HPLC. And at the end, we end up defining the parameters that are important for our model, which are the maximum growth rate in exponential phase, the maximum uptake rate, which is just the rate of how they saturate the uptake rate for high resource concentration. Then we have that another parameter that was very important for our model was the half saturation constant, which is the resource concentration supporting half maximum uptake rate. Then the duration of the lag phase, which is how long the bacteria remain in this non-growing state. And finally, the excretion conversion rate, which is a proxy of how much benzoate is excreted in the environment. And then what we did was to just use a set of differential equations in which we model the resources explicitly. And then in here, I'm showing those equations. So basically, we just model the bacterial growth, which is dependent on how much of the resources they are using. And then in the second set of differential equation, we have the changes in resources over time, which is related to the growth of the bacteria. And then with this model, well, I mean, I can spend a little bit more of time. So basically, as I mentioned before, these dynamics are really related to the maximum growth rate of the bacteria, and also is related to the half saturation constant, and also to how they are uptaking these resources over time. And then finally to the excretion rate of the benzoate. So the first thing we wanted to do with this model is to just validate the previous result that I showed. So basically, when we actually use this model, we do see that we can recapitulate the behaviors in monocultures, in co-cultures in benzyl alcohol, and also in citrate, which is the other competition. So we do see that with this model, we can recapitulate and capture these ecological dynamics quite well. So now the question was, what dynamics do we observe in fluctuating environment? So for example, in this case, we define one-day fluctuation environment means that one day we simulate that they grow in citrate, and then we transfer them in benzyl alcohol the next day and again to citrate. And that's what we simulate or fluctuating environment. So when we do this, we do see that when we have it in one-day fluctuation environment, the two species can coexist over time. And one species is more favored in one condition than the other. But then when we, for example, start increasing the length that they stay in each of the carbon sources, so for example, 2-day fluctuating environment means that 2 days they spend in citrates and 2 days in benzyl alcohol, we do see that we still have species coexistence, but hopefully you can start seeing that the more they stay in one condition, the amplitude between the two species start getting larger and larger. And then when we hit the 4 and 5 days fluctuating environment, we do have that actually Pseudomonas bring Acinetobacter to extinction. And this, in the model also, what we observe is that Pseudomonas eventually also end up going to extinction. And the reason is because it outcompeted the copartner that it needed to coexist in this environment. So in these 4 and 5 days fluctuating environment, we see a complete extinction of the community. So now we wanted to actually see if we can see these results with experiments. So what we did was to just grow the bacteria in co-culture, starting with a ratio 1:1. And we did this in triplicates for 6 days. And with experiments, we actually see again these dynamics that we actually predicted with the mathematical model. So we have that in 1, 2, and 3 days fluctuating environment, we have coexistence of these 2 species with the amplitude between the 2 getting larger. And then for the 4 and 5 days fluctuation, we have extinction of Acinetobacter. So we do not see extinction of Pseudomonas. We just see that Pseudomonas is actually to persist at very low densities when we have them in benzyl alcohol, and eventually with citrate, they just recover. So now that we define this part, so the conclusion is that fast fluctuation maintains species coexistence, but for example, in an environment that fluctuates slowly, what we can see is that we observe coexistence breakdown. So all of these that I've been talking about are ecological dynamics. So this is over 6 days, which is around 70 generations for these bacteria. So we assume that evolution is not playing a big role for these ecological dynamics. So the next question we ask is, what happens if we repeat this experiment, but now in evolutionary timescales? And then we were particularly interested about this condition of the cross-feeding interaction. So we wonder how stable is this cross-feeding interaction if we do it over evolutionary timescales. So for example, 200 generations. And the other condition we were very interested is the one day fluctuating environment. So in this condition, we saw coexistence of these 2 species. So we wonder if we can actually have coexistence over evolutionary timescales. So in order to perform this, we did a large-scale evolution experiment in these 3 conditions. So keeping just benzyl alcohol as a sole carbon source, just keeping citrate, and then in our daily fluctuating environment. And we did these experiments in monocultures and also in co-cultures. And then finally, we did for each of the conditions 4 replicates. And the whole experiment ran for a month, which is 200 generations. So the first question, so is cross-feeding interaction stable over evolutionary timescales? So here I'm plotting the result of just one of the replicates, which is consortium number 2. So you can see that now in the x-axis, I have the time now over 30 days, and in the y-axis, I have again the cell density. And then what you can see is that there's species coexistence over evolutionary timescales, and then there's definitely some kind of fluctuations in the cell densities over time, so it's not that constant. But overall, for the 4 replicate or 4 consortium, we see species coexistence until the end of the experiment. So we concluded that indeed this cross-feeding is stable over evolutionary timescales. But now, if we actually look at the second condition, which is the 1 day fluctuating conditions, now we do see a different case. So for example, here, again, I'm plotting just the result of one of the consortium number 2. And then here is very interesting because we see that at the beginning of the experiment, the 2 species coexist and maintain certain stable population dynamics, but then around day 14, something happened to one of the species, in this case to Acinetobacter, that it started decreasing in cell density until actually it gets extinct. And even more interesting, at least for an evolutionary biologist, is that actually this outcome of extinction of one of the species was only one of 2 evolutionary outcomes. So we do see that this extinction occurred in 2 of the replicates, but in the other 2 replicates, we do see coexistence of these 2 species until the end of the experiments. So then this is very exciting because this is definitely a different evolutionary outcome. So the one thing we can do with evolution experiment, which is very nice, is that we can freeze these bacteria at different time points during the experiment. And that's exactly what we did. So we kept a frozen record of these populations at every 7 days. So every week we will freeze these whole tubes with the bacteria. So what we can do then is to go back in the experiment, let's say at day 14 before the extinction of one of the species, and then revive these bacteria and see, for example, if we do a replay evolution experiment to see if we again see the extinction of Acinetobacter or not. And when we did these replay experiments, this time we did it with high replication. So we repeat this experiment for each of the consortium. We did 6 replicates. So what we do see is that for the 2 cases where we previously observed extinction of Acinetobacter, we again observe extinction of this species when we replay the experiment. And in the other 2 cases where we previously saw coexistence, we again recapitulate this coexistence when we do this replay experiment. So the conclusion is that extinction was a highly deterministic event. So it's not random at all. So we can definitely replay these dynamics. So then the next thing we wanted to know is what is causing this extinction of one of the species. And for answering this question, what we did was to isolate some single clones at different time points. So at the end of the experiment, at day 14 and at day 0, so which is the ancestor. So we isolated one single clone for each of the replicates of the population. And then we did growth curves experiments, and also we sequenced the full genomes to try to see if there were any genetic changes that could explain these deterministic dynamics. And first, I'm going to show the results that we have when we actually do the full genome sequencing the end of the experiment. So when we take this clone at the end of the experiment, we did the growth curves. So again, we just estimated the maximum growth rate and the yield. And when we do this experiment, I mean, here I'm just summarizing the results of all the experiments, but hopefully you can see that there's a lot of green, and the green indicate that there's actually a significant higher growth of the evolved types compared to the ancestral types. And these indicate that actually over the course of the evolution experiments, the evolved type are definitely growing better than the ancestors. So this is already a sign that these strains actually have adapted to these conditions. And then when we actually look at the genomes from this single clone, so what we did was to sequence the genome of the evolved type and the sequence the genomes of the ancestral types, and then we just compare the 2 genomes, and then we call de novo mutations, so anything that was not present in the ancestor and then that we observe in the evolved clones. And overall, we can say that we observe that we observe around 1 to 2 fixed mutations per clone. And when we look at where are these mutations located in the genome, we see that these actually mutations are not randomly distributed in the genome, but tended to hit certain genes. So for example, in Acinetobacter, we observe 15 mutations in this O-acetyltransferase gene. And then, for example, in Pseudomonas, we do see that they accumulate mutation in these 3 genes, which is a sensor protein, a flagellar component, and transcriptional cyclid D-GMP. So basically, we do observe a high level of parallelism, which already indicate that these mutations are most likely adaptive. And then also when we look how they are distributed, not necessarily in the genome, but among conditions, we do see that these ones tend to not be specific to one particular condition, but they were observed in many of the conditions. So for example, in the case of Acinetobacter, we do see this mutation in the condition of the constant benzyl alcohol, in monoculture and co-culture, but also in the citrate and in the fluctuating environment. And it's similar in the case of Pseudomonas. So overall, the conclusion is that species show signs of adaptive evolution after 200 generations, but these changes are not specific. So it seems to be general adaptation to the culture conditions. So now we go back to, now that we characterize these evolved types, so then we go back to this idea of trying to see what happened at day 14, which is just before the extinction of one of the species. And when we go back to that time and we do the full genome sequences, I'm summarizing in this table what we observed. So Pseudomonas, even after 92 generations of evolution, already have 2 to 1 fixed mutation in the genome. And again, this kind of mutation, we observe them in monoculture, suggesting their general adaptation to the culture condition. But I think what's really interesting is that in the case of Acinetobacter, we do see that in the 2 cases where it went extinct, we do not see mutation at this time point of 92 generation, where in the consortium number 3 and number 4, we do see that this Acinetobacter has 2 or 1 mutation. So our current hypothesis that we are trying to test is that mutations in Acinetobacter rescue it somehow from this extinction. And in particular, our hypothesis is the following. So we think that Pseudomonas accumulate mutations, so it's rapidly evolved, and these mutations actually confer higher growth to these Pseudomonas, and these exert an indirect effect on Acinetobacter, which either has to, it goes extinct, or it has to keep up with this higher growth of Pseudomonas, higher competition, and then it can actually, if it has adaptive mutation, then it can coexist with Pseudomonas. And this hypothesis is, again, sustained by this observation that Pseudomonas actually has higher growth after 92 generation. So we think it's really the mutation that is causing this higher growth. And also, the second line of evidence to support our hypothesis is that we went back to our mathematical model, and what we did was to input this growth that we observe after this evolution. And when we do input this growth into the model, so for example, in the case of the ancestor competing versus the evolved types of Pseudomonas, with this higher growth, we do see that Pseudomonas can actually, by just growing better, outcompete Acinetobacter only after 20 days. But actually 20 days is quite different for 14 days. So we believe that it's not only indirect effects, but there might be actually direct competition. There might be some antagonism or some, some kind of type 6 secretion system that Pseudomonas might be antagonizing Acinetobacter in addition to using more resources. So to conclude this part, we see that positive pairwise interactions are stable in a constant environment, that's the cross-feeding environment. And we do see that there's 2 outcomes in a fluctuating environment. So we either see coexistence or extinction of one of the species. And I don't have to actually give this take-home message to this audience because you're very well aware of this, but our conclusion is that the environmental context matters. So it really matters who is your neighbor and who is evolving next to you in terms of species. And then in that sense, we should move forward into not only study experimental evolution with single species, but try to do these evolution experiments in a community context, considering interaction with different species. And this concludes my talk. I left ample time for questions, and I just want to thank the people involved in this project in Switzerland. So my postdoc advisor, Martin Ackermann, Clément Villain, and Jean-Claude, who helped so much with the sequence. Also the whole lab in Switzerland, and the funding, which mostly came from EMBO and from the adaptation to a changing environment from ETH Zurich. And thank you very much for your attention.
Thank you very much for the nice talk. So we have time for questions. So if you have any, please use the raise hand tool or type it in the chat. And while we are waiting, I can ask one question since we have a lot of time. So have you tried to do a co-culture experiment where you put the Pseudomonas evolved in, or the Acinetobacter evolved in co-culture that coexisted with the Pseudomonas that actually outcompeted the Acinetobacter. So sort of to, well, probably technically it's difficult, but to isolate the evolved strains in different experiments and pair them together across different experiments.
Yeah, no, absolutely. That's that great one. So we did like a preliminary experiment with only one replicate, and unfortunately then the pandemic hit and we couldn't continue this experiment, because this is definitely the way when we can test this hypothesis. So evolving these clones. So what I can say is from our preliminary experiments, which I have here, so this is evolving, like competing these clones at generation 92. And actually from these pairwise competitions of the evolved clones, we do not see this extinction of Acinetobacter. As you can see, consortium 1 and 2, which is the one that Acinetobacter is supposed to go extinct, actually we do not see this. But this was with one replicate, and we definitely have to repeat these experiments, and also the conditions were not fully optimized to replicate the conditions from the experiments. But definitely we need to be trying to play with these evolved clones versus ancestors and these kind of replicate transplants kind of ideas to try to test these hypotheses. So far it seems like it's really at the population level and not at the clone level that we do see these behaviors. So we don't know exactly what's going on, if we didn't isolate the proper clone, or if it's something that is really characteristic to a whole population with whole like genetic diversity in some sense.
Yes. Yes. There is one question from Flavia.
Oh, yes. Can you hear me?
Oh, yes.
Okay, cool. Very nice talk. Congratulations. I'm interested to understand if you can measure somehow if these bacteria that you worked with, if they differ somehow in their mutation rate, or how they evolve.
Yeah, no, that's a very great question. So we definitely tested the mutation rate. So they have very similar mutation rates, these two species. So we thought for some reason that Pseudomonas might evolve faster, because that's kind of a common belief in the literature, but they are actually very similar in terms of mutation rate. And in many other ways, they tend to be similar species. So they have the same substitution rate, for example. So
Okay, yeah, I was curious because of that. Maybe I don't know if there is any other bacteria that you can select and make an experiment with this different rates of mutation, or I don't know, just
yeah, no, that's a very good point. Yeah, that would be super exciting to start tuning like this mutation rate, and to start seeing like how by tuning the mutation rate in either like very fast evolving versus slow evolving, like how would that play in all dynamics. And that's definitely possible, and that's definitely something I would like to do in the future.
Okay, I'll look for it.
Sounds good.
Great, we have time for more questions, if any.
Hi, I have a question. Sorry,
yes,
start my video. Thank you very much for your very interesting talk. I had one question I wanted to ask about the plot. When you've done longer time, like when you didn't look anymore, like when you wanted to look at the evolution, so you took longer time, I think up to 28 days. Maybe I didn't get it from the talk. Like, did you keep the subculturing time like at every day? Or
yes,
it was
yes.
in the batch? Okay. Yes.
Yes.
Okay.
So we didn't adjust anything based on how they start evolving. So we didn't adjust that. So we just for now, for simplicity, we just kept it content to 24-hour cycles, regardless of if they are first evolving faster and like they are like growing faster and saturating faster. Like we ignore that part for simplicity for now.
Yeah. And in the case, like, because at some point in this kind of experiment, when you see the abundance decreasing, of course, because they are going towards extinction, did you, like, is it possible to adjust the amount you subculture? Because of course, if you subculture, you're going to take a smaller amount of cells, and that can kind of a bit bias, like the direction towards extinction. Yeah.
Yeah, actually, that's a very interesting point, because we did the experiment starting at different ratio. So starting either at very high density, because we wanted to make sure it was not just stochasticity in the terms of just ecological stochasticity.
Yes.
So actually, we repeated this experiment, not in a ratio 1-to-1, but starting with a lot of Acinetobacter. And even starting with a lot of Acinetobacter compared to Pseudomonas, we still see
the same
this
trend.
decrease to extinction. So
Okay.
it seems it's definitely not this other kind of stochasticity.
Perfect. Yeah. And if I can ask, like, I mean, this is a further curiosity, because I really enjoy this topic. Like, are you thinking, or is it interest at all, like, to test co-fluctuations, like more than one parameter fluctuating? Because in this case, you apply fluctuation in the nutrient supply, but like, would it be of any interest to model and then apply fluctuation, let's say, in temperature or another, like, increasing the heterogeneity of the environment?
Oh, absolutely. So one of the experiments that, yeah, we're very excited is like to somehow try to add abiotic stress to this. So for example, yeah, temperature is a great like kind of suggestion and something we thought about it, and then try to tune a little bit more. Again, what would happen if we favor more one than the other? In this particular case, for example, what would happen if we favor the one that is not doing so great, like Acinetobacter? So yeah, definitely, in a way, I feel like this is just the start of setting up the system and playing with it. And now is the fun part, because we can actually start testing a lot of ecological and evolutionary theories and tuning stuff. And actually this system is nice because we can definitely tune interaction, evolutionary rates, and many things. We can also add more species if it's
yes. Thank you very much. Very interesting.
Yeah, thank you.
Great. There is another question from Aditya.
Hello. Thank you for the talk. So I was wondering, so I guess you showed here that when you have evolution in the case of a externally imposed fluctuating environment, it destabilizes the coexistence. But so we saw some talks earlier having to do with bacteriophage interactions. In this case, the fluctuations are internally driven by the dynamics of the different strains. And in that case, I think we saw something like that evolution can change the dynamics qualitatively by changing the direction of the cycles between bacteria and phage in phase space. But I guess, like, I'm not sure if I have a specific question, but I would just be interested in, like, whether you can comment on the difference between externally driven and internally driven fluctuations, and whether these are having, whether you think these might have qualitatively different effects on coexistence or not.
Yeah, no, that's a great point. So actually, I mean, reviewing literature, I feel like this interesting fluctuating dynamics is mostly given by these interactions that are either predation or like, you know, like where you have like more like a plus minus interaction, or you can have a plus plus interaction. But in this particular case, when you have just competition or commensalism, it's kind of interesting not to observe these internal fluctuations. It seems like it's really like this is it's more likely to happen with certain kind of interactions than with other kind of interactions. So in this particular case, yeah, we do not see anything of that sort. But
yeah, but do you think that do you think that exogenous versus endogenous interactions, or like would they qualitatively have different effects on coexistence? Is, I guess, my question.
Yeah, I don't know, like, it's hard to, I haven't think about it, to be honest with you. But I think that's a really great thing to think about. It's definitely like, it gives me like something to think about. I don't know how to answer, but it's a great point. It's really a great point.
Thank
What's
you.
your intuition?
Thank you.
Okay.
Great. If there is any more question, please type it in the chat or raise hand. I don't see anyone. Okay. So thanks a lot, Ale, again, for this very nice talk. Thank you very much for answering all the questions. And now we are going to be split again in the breakout rooms. And we are starting in about half an hour with the second lecture by Justin Eaker. So thanks, Alejandra, again, and thanks everyone for staying with us.
Well, thank you so much for the invitation.
Thank you.
Okay, welcome back everyone to this last session of today. We have now the second lecture by Justin Nickel. So please, Justin, you can share your screen and start when you're ready.
Great, thank you. Let me, one second. Okay, so I'll assume that you can see my screen. Thank you. It's a pleasure to be back. Today, I would like to focus on the role of humans in ecosystems and how we can understand that from reconstructing past systems. And so I'm going to spend part of the talk today focusing in general, really, well, more specifically to the Pleistocene with respect to the Pleistocene extinctions, and then focus in on a particular system that I've worked on in Egypt. So that's kind of the roadmap for today. Okay, so the effects of humans on ecosystems. We've been around for a while. The human species for 150, 200, 250,000 years, depending on where you make that break point. And of course, our relatives have been around much longer. I like to think of the process of our involvement in systems as a continuum because it has been, and our role in ecosystems has likely changed over the last 5 million years or so. That we began to diversify into different species, and then, of course, throughout our history as Homo sapiens. I'm really going to, even though I'm showing our ancestors or close cousins, I'm really going to be focusing on humans specifically. So let's start more recently than what is shown on the last slide and think about the Pleistocene extinctions. This is a period of time where we've come to understand the role of humans much better in the last number of years. But this is a period of time where we had a large influence on the global diversity, especially of mammals. And I'm going to focus a lot on mammals today, specifically larger bodied mammals, greater than 4 kilograms. So this graphic, let me walk you through it a little bit. It shows a lot of information, and it might be a little outdated. It's from 2006, and there's been a lot of work. This is a really quickly developing field. But what we're showing, of course, are different continents, and the numbers and pictographs of species relate to the number of extinctions that these different continents experienced as humans colonized them. The black bar represents when humans kind of showed up on the scene. Of course, in Africa, we've been on the scene for a very long time before we were Homo sapiens. And as we colonized the planet, we see a series of extinctions that we collectively call the Pleistocene extinctions. Some of these are very well timed with human arrival, and some of them are less well understood. So in Europe, for example, there's fewer extinctions, and our arrival is likely much earlier than the extinction than we have evidence for the extinctions being. The circles represent evidence for extinctions. Yellow is provisional evidence, green is robust evidence, and we have really robust evidence around 14,000 years before present. Humans showed up a lot earlier before that, so it's not as clear in Europe what's going on. In Australia, it's very, very well, I guess I could say clear, or maybe to be a little more careful, correlated, where humans arrive right as we find evidence for mass extinctions in Australia, and that's around 72 to 44,000 years before present. In North America, it's also very correlated, to use a more diplomatic term, where there's a large number of extinctions that we have evidence for right around the time that humans arrive on the scene. In South America, there's also a large number of extinctions, but it's less well understood. So humans appear to our understanding, humans appear to have played a large role in the Pleistocene extinctions across the planet. So what can we learn when we get down to business and we want to reconstruct how these ecosystems function before and after human arrival? And so that's really the goal today, to reconstruct patterns of interaction, to understand the role of humans on community dynamics over this period of time. What I'm showing on the right is an American lion skull. Lions are an interesting example of what occurred at the Pleistocene boundary. Lions were one of maybe the most widespread large mammalian carnivore on land. They were successful in many, many different continents. They were all over North America. The North American lion had larger body size than the modern African lion, which is interesting to imagine. And now, of course, their range is restricted to sub-Saharan Africa and a small population in India. And I want to focus a little bit more on how we go about reconstructing interactions in this particular way, because I'm a little more involved in this style of research. And really, the idea here is to take advantage of modern contemporary systems to understand the rules of interaction. So we'll use the Serengeti rules to kind of quote the documentary film and book on ecosystem processes. And use body size from contemporary systems as a window into what likely occurred in past communities, how past communities were arranged. I'm showing one of my two favorite graphics here, figures from a paper published, it's covered up by the window here, in the 2000s by Anthony Sinclair and Justin Brashares and Simon Maduma. On patterns of predation in a diverse predator-prey system. So this is in the Serengeti, and what you're looking at is, on the y-axis is mammalian predators arranged by their body size. So the smaller bodied are on the top and the larger bodied is on the bottom. Of course, lions at 150 kilograms are the largest terrestrial carnivores in sub-Saharan Africa. You know, if we throw in like crocodiles, they might not be, but we'll just focus on the grasslands in the savanna. And what we see is on the x-axis is the prey weight range in kilograms. So this is their food. And of course, as we go from the smaller carnivores to the larger carnivores, they're eating larger food. They're eating bigger food, right? They're eating herbivores that are larger, and that makes sense. But the other key thing to see here is the range. And that's what's highlighted by that black bar. So as we get to larger carnivores, they're eating larger ranges in addition to the larger body size, the mean. And it would be nice to see distributions on these bars, but I guess you want to plan your publication strategy. You don't want to throw it all into one go, because I think the data are out there. So lions have this huge range of food that they eat. You can see one of the interesting things here is wild dogs. They have this like shifted range relative to their body size. They're eating above their weight. And that's because they're such efficient pack hunters. So that's a pack hunting signal. But it's a nested relationship. So the diets of smaller predators are subsets of the diets of larger predators. Now, this means that if you're a small herbivore, and you live in the savanna, you're going to be eaten, and that's shown on the bottom graph. You're somewhat doomed. That's your future, is to be food, and only if you're larger do you escape predation. So, in the bottom graph, which is really striking, is the percent of mortality due to predation on the y-axis, and on the x-axis is the log prey log herbivore weight. I have to move around my little zoom bar that's in my way. And what we see is this very sharp sigmoidal kind of switch here, where below 310 kilograms, you're doomed. And above 310 kilograms, you've effectively escaped predation for the most part. And the dividing line appears to be zebra and buffalo. So the Z stands for zebra, wildebeest, they're eaten. Buffalo, and upwards, you escape predation. So buffalo, giraffe, rhinos, hippos, elephants, a very small percentage of their population is actively killed. We're not counting scavenging here. Okay, so bottom line, body size really matters in these terrestrial systems, in these mammalian systems. And we can really think of sub-Saharan Africa as being a remnant of the Pleistocene. It's kind of one of the last surviving Pleistocene systems for us to draw inference from and correlate our understanding of these systems to the past, especially a relatively recent past like the Pleistocene. Organisms were, it's not like we're going back to dinosaur world and trying to reconstruct which dinosaurs ate whom. You know, this is a little closer in time, so we can more confidently make relationships between what we see today and what we saw back then. Oh yeah. Okay, so this is how we're currently doing this, and it's based on a method published by Rudolf Rohr, I think in 2010, in American Naturalist, where he establishes a logit function where the log of the ratio of the probability that there is an interaction between a predator and prey divided by the probability that there is not an interaction between predator and prey is a function of the mass ratio between predators and prey. So here M sub I is the mass of a predator and M sub J is mass of a potential prey. And you can see that there's three different terms here, one of which is squared and one is linear and one is constant. And this allows, and by fitting alpha, beta, and gamma, which are unknown parameters that need to be fit to a particular system, by fitting these parameters, we can establish a Gaussian-like distribution in log space that defines the probability of a link existing between a predator and a prey as a function of the predator and prey's body mass in this case. So for example, by fitting, and this is, you know, fitting these three parameters could be done by a you know, any kind of you know, simulated annealing or Nelder-Mead you know, algorithm. So by fitting these parameters, for example, in the Serengeti, in the known Serengeti system, we can accurately predict 74% of link presence as well as link absence. And what I'm showing on the bottom here is not the Serengeti. It's the one that I had on hand when I was making my talk because I'm working on some ocean systems right now. But this is the Benguela system. This is from the famous paper by Peter Yodzis. And it shows predators and prey or species arrayed by their body size. And the known trophic interactions being the darker colors, the darker squares in this adjacency matrix, and the predicted probability of an interaction overlaying on top in the color. So by feeding in a known adjacency matrix, we can fit these unknown parameters, alpha, beta, gamma, and derive a equation that tells us the most likely, you know, the probability of an interaction given body size ratios. So by looking at the Serengeti, which I'm showing on the bottom, and for some strange reason, I've put large herbivores on the left and small herbivores on the right. My apologies. I tried to switch it around, but it wouldn't let me. It's a detail. What we see is by looking at the probability of an interaction or an established interaction in modern systems, we can equate that to the likely interaction structure for past systems, assuming body size rules stand. Okay, and there's not really a whole lot of evidence to suggest that past systems wouldn't be following the same allometric principles as modern systems, especially if we're within the same kind of families of organisms. But that's an assumption. Okay, so let me see if I can pull up my chat window just in case. Okay. All right. So not only do we get mean probabilities, well, this is just kind of showing a different cross section, right? So here on the bottom left, now I'm looking at, we have the probability of an interaction on the y-axis, and this is the mass ratio on the x-axis. And so you can see this. This isn't logged. The x-axis isn't logged, so it doesn't look Gaussian, but it is if you log it. And this just shows that for a given predator and prey, let's say if we keep the predator mass constant and we're moving the prey mass, there's a prey mass where the prey is too small for the predator. So it's energetically inefficient for a lion to run around trying to catch mice. But then it's also energetically inefficient and risks death to go after the largest animals as well. So there's these boundaries on either side. And this is what this is capturing, where those boundaries change as a function of what the organism size is, the consumer size is. Okay, so not to belabor, oh yeah, and then I always say not to belabor the point, and then I belabor it. But then when we expand this, looking at prey mass by predator mass, we can picture this in 2 dimensions, and you can see how the probability of a link is changing and captures that nested relationship that we see in the Serengeti. So this is one of the important things that this is capturing. Now, the original Rohr model also allows for latent traits, so traits not associated with body size. So, you know, thinking about marine systems, what, you know, sperm whales, or not sperm whales, that's a bad example, but humpback whales are not going to obey this relationship because they're going after really small things, and they happen to be really, really big. And so there are other latent traits that are not body size related that you could account for using this type of approach as well, that I'm not going to go into. Okay. So the questions that we want to address here is, how do changes in structure translate to dynamics? So if we take modern Serengeti systems as kind of the baseline and use it to reconstruct Pleistocene systems to get the structure of interactions, and really these are probabilities of interactions. So when I say reconstruct a Pleistocene system, I'm really talking about a large number of potential systems that fit those probabilities. Are there differences from one Pleistocene system to another? So this is really awesome work led by Machias Perez in 2015 in Proceedings of the Royal Society B. I really like this paper, where they implement this method. They reconstruct structure and using body mass ratios, and then just look at the structure of the system. They look at modularity by nestedness to see if there are differences between continents. Pleistocene systems from North America, South America, and Africa. And what they find is that there are large differences from one system to another, but it doesn't always divide out by continent. So for example, in South American system, there's a large range of modularity predicted by this approach, and a relatively constrained amount of nestedness that's predicted by this approach as well in all places. In North America, there's more modularity than in some of the Pleistocene systems than is seen in some of the Pleistocene systems from South America. And then Africa kind of falls in the middle here. It's in mid-range, and that's represented by this. I'm pointing at my screen like you can see that, but I should use my pointer, I guess. So African systems fall here somewhere. But Machias and his group went one step further, and they asked, well, since we can estimate the structure of these Pleistocene systems, can we assume some very simple dynamics and put those dynamics on that structure? So they used a Lotka-Volterra framework with allometric vital rates. Okay, so I'm just showing the very basic framework that they used. R sub I, and so this is, you know, the change in abundance for species I over time. And R sub I is positive if it's an herbivore, and negative if it's a carnivore. To capture that difference in growth and mortality. And in addition to the consumption of different prey species. And R is assumed to be an allometric, so it's scaled allometrically to mass to the -1/4. And this term B sub ij is extrapolated from these probabilities of a link existing between species. Okay, and you know, the system, it's a, you know, high dimensional ODE system, is stable if the leading eigenvalue is less than 0. And what they experimented with is simulating the system, the Pleistocene system as is, and with the presence of an added apex predator. So they did it without humans, and then they included humans, and they assumed humans had a similar hunting behavior or hunting focus as other apex predators in the system. So as, for example, a large lion or a saber-toothed cat. And there is a lot of evidence to suggest that humans were consuming at the top of the food web in the Pleistocene. And by looking at the system without humans and with the added effect of humans, they could assess the destabilizing effect of humans. Okay. So this is what they found. So on the top, I'm just showing the structure as before. On the bottom, the y-axis is the destabilizing effect of humans. And we have the different systems across continents according to the colors coded to the key on the left. And what we see is that in North America and South America, humans had a much larger destabilizing effect on the system, whereas in Africa, they had a much smaller destabilizing effect, okay? And this is suggestive that because, well, the idea here is that humans evolved in Africa, and they co-evolved with African systems. And it's possible that African systems, as we know them, are to some extent a product of those interactions. So the organisms that are present in African systems and the interactions that they have within themselves are a consequence of interacting with humans, in particular over the last 200,000 years or so. And this may explain to some extent why there have been so few documented Pleistocene extinctions in Africa relative to North America or South America, where humans were brand new on the scene. So those communities had no preparation for the arrival of humans. And that could explain the destabilizing effects that we observe when applying this type of theory to those communities. Just a very brief mention. So we're applying some of these same approaches to understand the change in the Northwest Atlantic over time. We're specifically focusing on the Nova Scotian shelf here, and that's circled in the map, and thinking about how that system might have changed over the course of the Holocene. Of course, that system began as a subarctic kind of icy environment, and is now not. In addition, human fishing has been a huge stressor on that system for hundreds of years. And we wanted to understand how that system may have changed as species were lost. And we're using the same types of approaches here. I'm just showing the same Benguela food web adjacency matrix that we're using as one of the food webs that we're characterizing the probability of links occurring between species. One of the challenges we've been having is many of the species in the systems that we're trying to parameterize, the contemporary systems that we're using as a baseline for this, it's very hard to find mass estimates if you're trying to do a complete system, you know, like what's a mass of a jellyfish. So we think so far it looks like these estimates work for length as well. So we could use, and that makes sense because it's a ratio anyway. So it's really whatever the organism is queuing off of to make its foraging decisions is what's important for this type of procedure. And just to give you a sense, this is what the Northwest Atlantic looks like today. We have some very charismatic species like the Greenland shark, which is one of the oldest organisms, animals in the world. We have porbeagle sharks, blue sharks, haddock, shrimp, capelin, all these interesting organisms. And this is what it looked like historically in the 1700s. It was very different. I've circled the organisms that are no longer large players in the system or extinct locally. We have Atlantic system, coastal sharks, white sharks, killer whales, walrus, cod, all of whose populations have either collapsed or gone away in the system that we're looking at. And so there's large structural difference between these systems, and we expect that there's going to be large dynamic differences as well. What's interesting is, you know, all of the fishing and managerial decisions that we make in the modern system here, the Northwest Atlantic, are basically working under the assumption that this is the system as it should exist. Whereas very recently it was very different. So the management decisions are contingent on our historical understanding of the system. And we think the historical understanding of the system isn't very good. So we're trying to add to that. Okay, I'm moving on now to Egypt. This is a beautiful palette at the Ashmolean Museum that's dated to the pre-dynastic era in Egypt. And what it shows is, among some mythical organisms, there's many, many modern species that are no longer in Egypt. So it's a portrait of the past, where the past is very different than the present. And Egypt in the pre-dynastic era was a very different place. And our goal here is to reconstruct the patterns of extinction, to understand what Egypt looked like before and use that to inform how Egypt operates today. And this is common. We have bared witness, sorry, as I hit the table, we have bared witness to these changes as humans, and we've documented these changes in many different places. This is a cave in Spain and a pictograph of a bison. Of course, there's cave paintings all over Europe and many other parts of the world documenting organisms that no longer live where they live, sometimes organisms that are extinct. And that's in the relatively short history of our being on the planet. The world has changed quite a bit, and we've likely played a role in it. Not always, and sometimes there's natural climate change reasons as well, but we've certainly played a role. This is another beautiful picture, the lion panel in a cave in France, I believe. I've not had the opportunity to see it in person, although many of you might have. But it's just incredible, not only just the images that are the artistry in the images, but they almost depict motion. And it's actually been thought that multiple pictures like this of lions next to each other were kind of a cartoon used to illustrate motion. Oh yeah, I wanted to delete this. Okay, so I'm just going to go a little faster here. Okay, so the goal here is to use paleontological and historical information to reconstruct the pattern of extinctions in a single community over millennial timescales. And specifically, we want to ask, what have been the cumulative dynamic effects of climate, urbanization, and industrialization on mammalian communities? And can this inform our understanding of how communities operate today? Now, I want to be really explicit in saying that I'm not trying to say what caused the changes that I'll be presenting. The causation is a different problem. Really, we're looking here at the consequence, reconstructing what those changes are and looking at the consequences of them. So let me just introduce you very briefly to the area. We're looking at the Nile Valley in Egypt. This is how it looks today. And I just love this description of the area. Desert vegetation can be classified into 3 basic subdivisions based on how much water those areas receive: perennial, ephemeral, and accidental. It's a very dry area. And water really drives everything. And if we look in Egypt today, these are some of the species we might be lucky enough to see. We have the black-backed jackal, Cainosaurus, or the golden jackal, sorry, hyena, the striped hyena, caracals, these really cute small cats, foxes. And if we were to go back in time, we would have seen other organisms as well. We would have seen leopards, we would have seen cheetah. These organisms are no longer there. The last individuals were killed relatively recently, although their populations collapsed a long time ago. The last leopard was observed in 1913. The last cheetah was killed in 1974 near El Magra. If we look at herbivore species, we see, of course, a little more diversity. We see the wild ass, we see ibex, gazelle, multiple species of gazelle, many of which are red listed. And then we look at those that are no longer there. The hartebeest was last observed in 1935. The Egyptian boar, the last specimen in Egypt was British specimen number 2450, 1912, Addix, 1957, and oryx, at the bottom, disappeared in the first half of the 19th century. However, if we look farther back, humans, keen observers of the natural world that we are, have been recording our observations for a long period of time. So our goal here is to not only integrate paleontological information of species occurrences, but integrate artistic representations of species occurrences as they have been depicted in Egyptian artwork over time. And because Egyptian antiquity is so well documented and dated, dating artwork and the presence of organisms in artwork is relatively straightforward. And so the idea is to use these representations, many of which are in ecological settings. So, pictured here is the enjoyments of hunting. And we see, herbivores being hunted by dogs, but herbivores in their natural environment being hunted by Egyptians. And what we're going to capitalize on are these ecological depictions of organisms rather than religious iconography. Here's again a beautiful pictograph of from the tomb of Amenemhat in Dynasty 12, picturing many different species of herbivores being hunted with a net, which was a common way of you surround an area with a net and then hunt them at ease. Hartebeest, Dorcas gazelle, leopards, oryx, fox, cheetah, roan antelope. So just incredible diversity, most of which are not currently in Egypt today. Again, here's a couple different pictures just to show you the diversity of in natural history in some of these depictions. We have bird hunting in the upper left, and the nets that, or the traps that they used in the upper right, and then the fish diversity on the bottom is also really kind of astounding. I don't know fish very well, but I would bet a lot of these fish could be identified pretty well from these pictures. Now, one of the other important things about Egyptian artistry is that they were very focused on documenting. They documented a lot of details, and they clearly distinguished organisms that were domesticated and imports from other places that might have been captured and paid in tribute to a pharaoh or whatever was going on. And this is the symbol on the bottom that symbolized domesticated organism. It's pronounced Ren, and it means fattened. And they use this to distinguish between wild and domesticated stock. So just a really quick and really brief history lesson on the Egyptian past and what I really want to focus on, which is the climate change. So 15,000 years before present, we have the oldest known artwork out in the desert, and I'm showing that pictured on the sides of these rock cliffs. Now, 5,000 years before present was the end of the African humid period. So before that period in time, Egypt was like East Africa. It was wet, it was a savanna woodland, and it had all of the organisms that we associate with savanna woodlands, which I'll show in a moment. But at around 5,000 years before present, the monsoon shifted and the rain stopped falling on the area, and it began its desertification process. Of course, that had nothing to do with humans. 4,580 years before present, we have the establishment of the Old Kingdom. 4,140 years before present, we have the establishment of the Intermediate Period, along with a large aridification event, which I'm showing with the orange dots, by the way. And this aridification event was an important one. It's noted, it's discussed in tablets and pictograms, and it's been linked to, this aridification event has been linked to large political upheaval and quick successions of rulers. And it's thought that quick succession of rulers was brought about by large famines from the aridification. 3,270 years ago was the establishment of the New Kingdom. And then 3,000 years before present was another large aridification event that's documented in the records, as well as sediment cores. Now we can confirm a lot of these aridification events. And then more recent history with the Greco-Roman description of Egypt, and of course, more recent industrialization. Okay, that was a very quick history of Egypt. Just to show you some early Holocene rock art from the Egypt area. We have elephants depicted up here. So we don't usually associate elephants with Egypt. They were there. Giraffes were in Egypt. The Domadeer was likely a recent migrant from Mesopotamia, and just a whole suite of organisms that we don't think about living in Egypt today because they don't. And if you go through and categorize all of the different species that are present both in archaeological or paleontological deposits as well as those represented in ecological settings, this is the list that you have. You have cob, wildebeest, hartebeest, oryx, camels, deer, domadeer, giraffes, hippos, just everything we associate with the Serengeti was in Egypt. In terms of the carnivores, we had striped hyenas, spotted hyenas, cheetahs, leopards, two distinct species or subspecies of lion, a short-maned lion and a long-maned lion. And I'll get to that in a minute. And that was distinguished apart from being male and female. And there's, well, I'll get to it. I'm getting ahead of myself. But if we go through time and document when these organisms disappear from the these ecological depictions and from deposits, we can build a pattern of extinctions, and we can put error on that too to account for organisms being depicted after they've gone, right? And so what I'm showing here is the first appearance and for some organisms, but primarily the last appearance of these different species, which is the black circle. And then the red, the color that's overlain on top is the probability of extinction. So where we have a last appearance, we have a gradually increasing probability of extinction following their last appearance, okay? And so it's a very particular pattern. And so that is what we used to infer changes in food web structure over time. Now, I mentioned that there's two subspecies of lion, a large-bodied long-maned lion. I gotta keep an eye on the time because I get trapped in Egypt stories here. A large-bodied long-maned lion and a smaller-bodied short-maned lion. And these were represented as two distinct subspecies within the artwork and descriptions. And this actually correlates with a known lion population that was last seen in the Atlas Mountains. So we think this might represent the Barbary lion, which was a larger-bodied, longer-maned lion that existed in North Africa until relatively recently. There's still stories of relict populations surviving in the Atlas Mountains, but it really disappeared at the end of the Second Dynasty in terms of its representation in the artwork. And then, of course, the short-maned lion lasted much longer. So it didn't disappear from Egypt until the end of until around 3,035 years before present, actually correlating with an aridification event. Okay, so one way that we can depict structure over time before we even begin to reconstruct interactions is to just simply look at the predator-to-prey ratio. So what we see over time, and I would also draw attention to the fact that these time bends are not equally sized, okay? They really correspond to the information that becomes present due to different dynastic cycles beginning or ending in antiquity. But if we look at the predator-prey ratio over time, we see that it's increasing until about 3,000 years before present as herbivores disappear. So herbivore species are disappearing first, increasing the predator-prey ratio, after which it decreases at around 3,000 years before present, and then increases again. Of course, as the system becomes, as it loses species, it becomes much more volatile. The predator-prey ratio becomes more volatile because smaller changes make larger differences in the ratio. But I'd like to draw your attention to three larger shifts that we see in the predator-prey ratio, at least historically, and that's outlined by these stippled lines. And those correspond to the three large aridification events. So it would seem in a very correlative way that aridification events appear to have impacted how the structure of the community may have been changing over time. And again, I'm not really drawing causation to this. There's many different causes that we could imagine could feed into this. And this might be, room for theory in the future. It could be driven by bottom-up forcing. It could just be driven by, changes to the environment as directed by changes to the climate, which I'm showing on the right. Or humans could play a large role. We could play a role by competing for space. Really, in this area, as it becomes more and more desertified, water is the key. And finding habitat where you have access to water is really the limiting resource in these systems. So as humans begin to expand their agricultural base, they're taking up space where water is available. And that's pushing out wild animals. And this could certainly have led to many of the extinctions that we observe in the record. Or humans could have actively been hunting these organisms and had a large impact, especially as we have the establishment of a relatively sedentary human civilization for one of the first times where they're also subsisting on crops for food, they could have an abnormally large impact on populations of wild organisms. Okay. Now, I'm not going to go through this because I already did, but we use body mass information because all of these species are extant. So we know the sizes of these organisms, and from their body sizes, we can reconstruct the probability of a link existing between them and then build series of food webs that we think best represent these systems. Dynamics. Okay, so this is kind of the fun part, and I know you guys have had awesome talks about dynamics so far. We wanted to not only reconstruct food webs, but say something about the stability of those systems. And we wanted to see how stability or measures of stability might have changed over time. To do this, we used an approach called generalized modeling. And I've got a couple key citations on the bottom. This was really pioneered by Thilo Gross in 2006 and later in 2009 with and then we have a description really focused on ecological systems in theoretical ecology in 2011. And I've been meaning, I'll try to make a reference list for all of these things that I'm citing. I'm not doing a very good job of reporting what journal they're in, but I'll make a reference list for all of the different things that I'm talking about for those of you who are interested that I can make available. Okay, generalized modeling. So, this is really useful where we know the architecture of the dynamics of the system, but we don't know the functional relationships that are embedded within that architecture. So for example, let's consider an ecological system where we have the change in biomass over time. So B is biomass. And we have some function of growth. So we have a source, right? So there's biomass is growing by the function S, and it's draining, or there's a function of mortality as this function D. But we don't know the specific nature of these functional forms. This actually might better represent our knowledge of the system. And this is particularly true for past systems where we really don't expect to know the exact architecture of the system, or at least we don't want to assume that we know. The problem is, of course, we can't simulate a system like this, and we can't solve for its fixed point or steady state. So what can we do with it? We can do quite a bit, actually. The method, very simply and generally and quickly, is to establish B star as a variable representing all internal equilibria of the system. So these are all non-negative, non-zero equilibria that represent states where all of the organisms persist in our system. Okay, but it's unknown because we don't know the functions. We can then define a new set of parameters representing the normalized variables of the generalized system. So here we're defining little b as the biomass divided by the biomass at steady state, which is unknown. A little s of little b is the growth function of the biomass divided by the growth function of biomass at the steady state. And little d of little b is the mortality function divided by the mortality of the biomass at steady state. The important thing about all of these new parameters, these normalized parameters, is that steady state, they're all equal to 1. And that helps us a lot. This allows us, this normalization procedure of the generalized system allows us to extrapolate biologically meaningful, and this is the key, biologically meaningful and relatable coefficients, terms, variables, etc. So here we see that when we set the system equal, so when we set the normalized system equal to 0, right? So assess the system at steady state. Little s of b and little d of b, I'm getting covered up here. So this term and this term become 1. And we have this simple relationship of s star over b star, which is just a constant, is equal to d star over b star, another constant. And we'll just define this as the turnover rate at steady state. So this gamma here is directly biologically meaningful. It is the biomass turnover rate at steady state, and it's equal to the normalized function of growth minus the normalized function of mortality. Okay, so we've taken this general system and we've normalized it, okay? Now we want, and then we've redefined some of the constants in the system to be biologically meaningful. But now we want to assess the system's stability. And what's our interpretation of, what can be our interpretation of this? So we're going to perturb it, and we're going to look at the conduct linear stability analysis as we would to any other ODE system that we might investigate. So we're going to look at the change in d little b over dt as a function of little b, with respect to little b, and this equals the single eigenvalue of the system. And we simply take the derivatives with respect to little b across the different elements of the system. So we end up with this derivative, for example, for the growth function, for the normalized growth function, with respect to the normalized biomass, is equal to the derivative of the log of the unnormalized growth function divided by the derivative of the log of the unnormalized biomass evaluated at the steady state. And this actually has a direct biological interpretation. It is the percent change in growth divided by the percent change in the argument and the unnormalized biomass. It's a functional elasticity. So functional elasticities in this system are the logarithmic derivatives of the unnormalized functions relative to the unnormalized argument. And this provides a nonlinear measure for the sensitivity of the function to variations in biomass. So we have, in this very simple 1D system, we have the single eigenvalue of the system equal to the biomass turnover rate times the elasticity of growth minus the elasticity of mortality. And we can directly relate this to the conditions under which this single eigenvalue is going to be greater than 0 or lesser than 0 as a function of the values of the elasticities of growth, the elasticities of mortality, and the biomass turnover rate, which doesn't matter so much as in terms of determining the positive negative value of the eigenvalue. Okay, so one of the important messages here is that, or why are we doing this, is that elasticities characterize whole families of functional forms. So consider power law functions. We have a function where it's constant, a function with a linear power, a function with a square term. And if we take the elasticities of these functions, we see that the constant term is simply 0, the linear term is 1, and the squared term is 2. So power law functions have direct single values associated with their elasticities, with their functional elasticities. And this allows us to set realistic bounds to the elasticities in the Jacobian, if we're talking about a multidimensional ODE system, that go into the Jacobian. So you'll have a Jacobian matrix that you derive to look at the linear stability of the system. That's a function of all of these elasticities from the generalized model. But the elasticities themselves have very small bounds associated with them to be biologically meaningful. And that really sets a lot of constraints into the system, so you can more efficiently search across combinations of parameters that are biologically realistic and get a better sense of what the stability of biologically realistic systems is, given the architecture fed into the generalized model. More complex functions have elasticities that are functions of the unknown steady state, but also range between small ranges of values. So for example, an elasticity that's more complex, that might be a function of the unknown steady state, may only realistically be able to vary between 0 and 1 or between 1 and 2. And this really sets lots of constraints that enables us to more effectively search biologically reasonable space. So just to kind of depict this as a cartoon, if we're doing a random matrix type of approach, we have a large parameter space to search across to get a sense of the stability of the system. However, the generalized modeling approach allows us to subdivide this large parameter space to biologically meaningful units, so we can more efficiently search across the system and ignore parameter combinations that are biologically unrealistic. Okay, so now that we've done this, we have, I'm pointing at my screen again. We have a large system of many interacting species. And we have, so change in the abundance of species I over time is equal to the growth of I if it's an herbivore, plus the growth of I if it's a carnivore consuming many other herbivores, minus the mortality of species I if it's a carnivore, or minus the predative loss if it's an herbivore. We normalize that system to a steady state. So this is just kind of the higher dimensional version of what I've already shown you, but all of the principles are the same. And then we can derive the Jacobian matrix. So we can derive the on diagonal and off diagonal elements of the Jacobian matrix, which are now all functions of the functional elasticities, each of which has relatively constrained ranges. And so now we can search across many different potential Jacobians by randomly drawing from within those ranges to determine a percent of those systems that are stable relative to a percent that are unstable. And this allows us to translate the structure that we've reconstructed from the body mass ratios to the likely dynamics of the system, and do so for all of the different snapshots that we have of Egypt in the past as it changes over time. So we are really going to look at 3 different measures here. We're going to look at food web stability, which is just the percentage of drawn food webs that are stable out of the large number that we simulate. I think we simulate around 10 to the 7th. And so this gives us a sense, again, of just the general stability of the entire system. Is the system stable, right? And that's just what percentage of those systems, those drawn systems, those Jacobians that we draw from the functional elasticities have a leading eigenvalue that's less than 0. Secondly, we wanted to assess the species-specific roles and sensitivities to change. Okay, so let's see. Yeah, so we did 2 things, which I'll describe in the next slide. So one is related to the stability of the system, and that is, once we have the stability of the system, then we can start pulling out species and determining whether or not that stability increases or decreases with the absence of a given species. So for example, if 90% of our food webs that we draw are stable, and then I pull out lions, and now 95% are, well, then that means that lions are a destabilizing force in that system. Okay, if we have a system that's 90% stable and I pull out gazelles and suddenly there's 10% that are stable, then that would mean that gazelles are an incredibly stabilizing force in that system. So that's something that we're going to look at. We're going to look at the change in the percent of stable food webs relative to different species being present or absent in the system. The third thing that we're going to look at, I don't even know if I have a 3 on here. I should. Do I? No, I don't. Okay. The third thing that we're going to look at is the sensitivity of species to a perturbation, to a different type of perturbation. So we're going to use this idea of a press perturbation, where we push the system to a different equilibrium, to a different steady state, and examine how each of those species that are in the system respond to that push. And this is given by the equation in the bottom right. And it's a function of both the eigenvectors and the eigenvalues in the system, where we sum across the modes to get the stability, or sorry, the sensitivity of each individual species in the system. Sorry, I'm going through this really quickly. We explain it more slowly in the paper. I guess as slow as you'd like to read it. So what are our results? What were the dynamic consequences of community change over time? So on the y-axis is the proportion of stable food webs with 100% at the top. And on the x-axis, we have our time bins. And so we look at each system and simulate many different iterations of its structure, as well as the potential dynamics that can go onto that structure. And what we see very straightforwardly is over time, the system becomes less stable. And this is true whether we apply tons of uncertainty into the disappearance of species. So, upwards of, plus or minus 200 years, plus or minus 500 years to the disappearance of species from the both the artwork and the paleontological information, archaeological information. It doesn't change the overall picture of what occurs as species go extinct. So the Egyptian system at the end of the Pleistocene, we can interpret as being much more stable as it is today, as it has fallen apart and unraveled over the course of the Holocene. Perhaps a little more interestingly, we can evaluate the stabilizing effects of different species in the system. So again, on the x-axis, I have the time bends. And here we're trying to determine whether there are key species that contribute to the stability of the system, in which case, if they're lost, they are contributing destabilizing forces into the food web. Now, on the y-axis, we have the change in the percentage of stable food webs with or without a given species, and each line, each trajectory represents a comparison of the presence or absence of different species. And I've illustrated some of the species. It turns out all of the herbivores are stabilizing. So, and meaning that their trajectories are going to be above zero because they have a net positive contribution to the stability of the food web, whereas carnivores tend to be destabilizing. And what we find is that the most stabilizing species are the smaller bodied herbivores. And as we've lost the redundancy of smaller bodied herbivores over time, their stabilizing effect of on the system has increased, which means the consequence of losing them has become larger as well. We can see farther back in time, losing any species has very small effect on the stability of the system. Whereas today, because there's so few organisms left in the system, losing any species for the most part has a much larger effect, but it's the smaller bodied herbivores that have the largest effect because they're shared by so many carnivores. Because the carnivore species rely on those herbivores, losing them is losing the core of the system. And the last result that I'd like to share with you has to do with the sensitivity. Does sensitivity predict persistence? I mentioned yesterday that one of the keys about paleoecology is that we know the future when we look into the past. We know the future when we look into the past. So we can evaluate whether species that we think might be likely to be important have large effects on the system. Or perhaps we can consider whether species that appear to be more sensitive based on some theoretical argument actually go extinct. So here we're calculating the sensitivity of species to a perturbation. In other words, we're pushing the system to a new equilibrium and seeing, how different species react. How sensitive are they to that disturbance? And we could postulate then that species that are more sensitive to a disturbance may be more likely to go extinct. In other words, species that are more sensitive to a disturbance may persist for a smaller amount of time in the system. And because we know the persistence of species, how long they are in the system after the end of the Pleistocene, that's what we have on the y-axis. We have how long the species are in the system after the Pleistocene. It's logged. And then we have the log sensitivity on the x-axis. And what we find is a negative relationship. So species that are more sensitive have shorter persistence times in the system, whereas species that are less sensitive have longer persistence times in the system. And there's this kind of roof here, the ceiling, because that's the maximum amount of time since the end of the, since we began making the records. So at the end of the Pleistocene until now, this is kind of the maximum. These species are still present in the system and these species have been lost. So what have we found? We found in Egypt, as the system has unraveled in the presence of all of the changes that have gone on in Egypt, both climate, human-induced, everything added together, we found that with the unraveling of the food web has come decline in stability. And that there are key species that have played a larger role than others in determining whether that stability is eroded or not. And finally, the sensitivity of these species, which is again a consequence of their structural relationship within the food web, as well as the elasticities that we attribute to them through this generalized modeling perspective, that the more sensitive species are, the less likely, the less length of time they are expected to remain in the system. So persistence in this case is predictable. Okay, so moving on into the future, right? We think these community level frameworks are vital for understanding how our ecosystems are expected to change in the face of these traumatic events that they witness over long periods of time. And of course, you know, we've looked into the past at mass extinctions in the past, and we've seen large changes in food web structure and function on either side of extinctions. And today it's important to note, not to end on too sour of a note anyway, but that we're in the sixth mass extinction. All evidence points to the fact that we are in the sixth mass extinction. And so building upon paleontological insight, I think, in my perspective, is vital, absolutely vital for having a sense of what to expect in the future. Yeah, so if there's any time, I'm happy to take questions, and then on Thursday, I'll really switch gears and take a much more a close view of how energetic constraints can give us insight into systems that are no longer around, that are extinct. So thanks so much.
Thank you so much, Justin, for the very nice lecture. So yeah, sure, there is a bit of time for questions. So if anybody has any question, you can either raise your hand or write it in the chat as usual. I guess everybody is a bit tired after many hours of lectures. Anyway, Justin will give another lecture on Thursday, if I'm not wrong. So if you get any questions later, you can always ask them on Thursday.
Great. Sounds great. Well, thank you, everybody. I appreciate it.
Oh, actually, there were raised hand. I didn't see them.
Oh.
Sorry. So there's a question from Flavia.
Hey, Justin, how are you?
Good. Hi, Flavia. How are you?
Very nice talk. Congratulations. Thank you for that. I'm curious with one result you presented. You showed that the small herbivores are more or less the keystone species. And this is a little bit contradictory. I don't know how to say that, but it's like the opposite of the expected. In theory, usually we think that large mammals or large predators are those that are the keystone species. Is your result a consequence of the dynamics or a consequence of the biomass approach that you use? What is that?
Yeah, you know, I think, and I wouldn't necessarily say they're keystone species, because that's assuming, you know, some kind of stabilization relative to their known abundances. But yeah, they're key to stability, but I would say they're only key to stability in the systems that we were looking at recently. So it's only after the systems are unraveled that they really become that important. But if you look at a fully fledged system, so if you go back to the end of the Pleistocene, which is very Serengeti-like, they, you know, there really aren't any species that make a large, that have a large impact on stability when they fall out. And so I guess I would argue that that is an artifact of the unraveled state of the system. And that perhaps only very disturbed systems will observe that importance for the smaller bodied herbivore. But, you know, even in these large systems, and I think that's because of redundancy. You know, if you look at that, that mortality graph in that Sinclair paper showing that sigmoidal relationship to the probability of predative mortality, the smaller animals are getting hammered, but there's a lot of them. And so if one goes extinct, you know, I can't find a topee, I'll go after Thompson's gazelle. But if there's only Thompson's gazelle, then I think that would be a very different situation.
Okay, I see. Thank you.
Okay, there is another question from Matteo.
Hi, yes, just a curiosity. just to what extent you can think the sixth mass extinction is similar to the others? Because essentially it looks like there is a factor which is the prevalence of humans, which is might be different from the other extinctions.
Yeah, that's a really good question. And I'll be wading, and I guess to answer that, I'll wade into things I don't know a ton about, because a lot of people are working on this, right? Just determining whether or not we're actually in a mass extinction, which has a lot to do with figuring out extinction rates and projecting into the future, into a very unknown future. I guess if anything, I would think that the rate of the extinctions that we're experiencing today is probably a lot faster than the rate of extinctions experienced in the past, which are kind of unknowable. You know, the Permo-Triassic mass extinction likely occurred over hundreds of millions of years. Or sorry, not hundreds of millions of years, you know, millions of years, or hundreds of thousands of years. And because it was this kind of gradual change to the climate brought on by these massive volcanic events, the KT extinction might have been a lot faster, the asteroid impact, but you know, we don't really know how the extinction rates changed for different taxa afterwards. But it, you know, we've had a huge impact on a relatively short time scale. And I would say if anything's different, it's faster. We're more efficient than volcanoes. So in terms of, you know, contributing to extinctions. So that would be my guess. But I know a lot of people are working on trying to understand how the current mass extinction, if it is a mass extinction, and it does seem like it is, compares to prior extinction events.
Thanks.
Okay, I still see the raised hand of Flavia. I don't know if she has another question or if she didn't unraise her hand. Okay, it was not a question. So I don't see any other raised hand. So if nobody has any other question, or if I am not seeing them like before, I think we can say thank you again to Justin, and hear you again on Thursday.
On Thursday, sounds great.
Have a nice evening.
Thank you. Well, you guys have had a long day, but my day's just beginning, so
Right.
that's why I have energy.
Have a good day to you then, and to all the other people on the other side of the world.
That's right. Thank you so much.
Bye.
Bye-bye.